Good morning. It is proud privilege for me to initiate the 2 days international conference on virtual mode. I would like to welcome the today's guests and distinguished honorable registrar and my beloved faculty members of botany department and participants from various nooks and corner of the country. Here it is a proud moment to celebrate the International Year of Health declared by United Nations 2020 as International Year of Health with the slogan protecting plants, protecting life. Here I would like to welcome all the dignitaries, Dr. Mahtab Bamji, Madam, Deputy Director Grade, Retired, National Institute of Nutrition, Honorable Registrar, Professor Naseem Man, Telangana University. I welcome one and all for the inaugural session. I request Professor Vidyavardhini Madam to address the gathering regarding the international conference on virtual mode. Over to Vidyavardhini Madam.
Thank you, Haleem Khan. Good morning and greetings to each and everyone from Professor B. Vidyavardhini, Telangana University. A warm welcome to Professor Naseem, Registrar, Telangana University, Dr. Mahtab Bamji, who's presiding her overview from the conference from NIN Hyderabad, all the eminent resource persons, the family of Botany Department, Telangana University, comprising of Dr. M. Aruna, Dr. A. Haleem Khan, Dr. Srinivas, Dr. Jalandhar, and all the participants. The two-day international conference on emerging trends in life sciences, ICELS, on virtual mode is being organized by the Department of Botany, Telangana University on the 15th and 16th of December 2020. It comprises of one inaugural session and 4 technical sessions with eminent speakers across the globe, delivering talks on various aspects of current trends in life sciences, as it's an established fact that life sciences plays a very pivotal role in the current scenario. And Edward Abbey aptly stated that we must learn to think not logically, but also biologically. And hence the international conference focusing on different aspects of life sciences with renowned speakers is being organized. We have Dr. Shyam Kumar Bandari from the University of Alabama, USA, who's speaking on small extracellular vessels, the next big thing. We have Dr. Santosh Kumar JU from Chulalongkorn University, Bangkok, Thailand, who's speaking on medicinal plants and their trade, how safe are raw herbal drugs. And we have Dr. Insaf Ahmed Qureshi from the Department of Biotechnology and Bioinformatics, School of Life Sciences, University of Hyderabad, who's speaking on development of novel multi-epitome vaccines against SARS-CoV-2 using immunoinformatics approaches. And the technical session 2, that is tomorrow, we are having Dr. Biju Dharmapalan from the CSIR Discard New Delhi, who's speaking on molecular biology for understanding complexities of life. And we have Dr. M. Tippaswamy from Davanagere University, Davanagere, who's speaking on RT-PCR and its application in croplands. And we have Dr. Harish Handral from the National University of Singapore, who's speaking on stem cells and additive manufacturing, the next generation of pharmaceutical screening platform. Thus, we have got eminent persons across the globe who are going to present their views on the emerging trends and the different aspects of life sciences. And I hope all the participants will have a good time, and we'll have, and hope you give your feedback, and hope you enjoy the sessions. Good day and God bless. Over to Haleem Khan, sir.
Thank you, ma'am. Now I request Dr. Aruna Madam to give her address regarding the inaugural of this session. Aruna ma'am, over to you.
Good morning to all the dignitaries and delegates attending today's webinar. On behalf of Department of Botany, Telangana University, I deem it as a privilege to welcome you all to this two-day international conference on emerging trends in life sciences during December 15 and 16, 2020. In this context, we have invited eminent scientists and speakers from all over the globe who are going to discuss and address novel challenges and emerging researches in multidisciplinary areas of life sciences. The purpose of organizing this virtual mode of two-day seminar during pandemics is to bring all the academicians, industrialists, professionals, researchers, and students onto a common platform to share their knowledge and to gain their experiences and also to have their innovative ideologies in research, and it's for betterment of human life. And life sciences is a broad area in the present era of scientific boom. From the origin to evolution and development of living beings, this subject traces all the traits and discoveries and discovers theories that aid the human race and environment in diverse manners. And the deliberations of these 2 days would determine the key trends of life sciences in various areas of expertise research in fields of biotechnology, nanotechnology, immunology, and physiology of crop plants, advanced techniques and principles of bioinformatics, medicinal botany, therapeutics, nutraceuticals, and so on. So with this, hope that all the participants and delegates will enjoy the seminars, that is lectures delivered during all the technical sessions where Professor Vidyavardhini Madam has given a brief detail of each speaker along with their topic. Hope that the interactions and discussions of the webinar will definitely motivate researchers, scholars, and students and ameliorate the skills and scientific temper of all professionals who are going to be with us for 2 days. Thank you, and thank you for giving me this opportunity. Thank you, sir.
It's a time to introduce our university and the department. Telangana University is established in the year 2006 by then the government of Andhra Pradesh to cater the academic pursuits of background and rural student community. Due to the inadequacy of facilities for higher education and to serve the needs of postgraduate and research activities in remote and inaccessible tribal areas, keeping in view to eradicate the regional imbalances of higher education in the state, Telangana University was established. Then the campus was operating in the Giriraj Government College, and in 2009 the building has been constructed and shifted towards the main campus in the Dichpalli, which is very near to the national highway crossing the Nizamabad district. So the university has got 577 acres of land with different buildings of University College of Arts and Science, University College of Computer Science and Engineering, University College of Commerce and Business Management, University College of Law, and a well-equipped library with 24 hours internet connection and hostels of three, one for women and two for men. And the university also operates in two different areas like South Campus in Bikanur, and in Sarangapur there is a center for education which offers you undergraduate and postgraduate course of education. Presently, the university offers different disciplines of science like biotechnology, botany, geoinformatics, computer science, statistics, chemical and physical sciences. About the Department of Botany is one of the pioneer department of the Telangana University, established in along with inception of university in the 2006. The department offers master's course and PhD. The master's course of botany is offered with a specialization plant physiology. The faculty members laid impeccable foundation of teaching and research among the peer group, and since the inception of the department, the Laurel faculty has been working on various research projects supported by SERB, DBT, UGC, and many other research organizations. Presently, we are having Kothari Fellowship Research Scholar work, postdoctoral fellow working, along with many of the students with Inspire Fellowship and Rajiv Gandhi Fellowship in the department. Number of scholars have been awarded PhD, and presently 16 scholars are working in the various fields of plant sciences under the able guidance of various research supervisors of the department. With these few words, I conclude my about the department in university, and I request our Honorable Registrar Madam to give her address to the gathering. Over to Registrar Madam, Naseem Madam.
Thank you, Dr. Haleem Khan, sir. Our esteemed guest of honor, Dr. Mehtab Mamji, Madam, Emeritus Scientist, retired Deputy Director from NIN, on the eve of the two-day virtual international conference on emerging trends in life sciences, organized by the Department of Botany of our Telangana University. Distinguished eminent resource persons, Dr. Shyam Bhandari, sir, from University of Alabama, US, Dr. Santosh Kumar, sir, from Chulalongkorn University, Bangkok, Dr. Harish Handral, sir, from National University of Singapore, Dr. M. Tippeswamy, sir, from Devangir University, Dr. Insaf Ahmed Qureshi, sir, from University of Hyderabad, Mr. Biju Dharmapalan, sir, from CSIR New Delhi, convener of this event, Dr. Haleem Khan, sir, head of the Botany Department of our Telangana University, staff of Botany Department, Professor B. Vidyavardhini, Madam, Dean, College Development Council of our Telangana University, and Dr. M. Aruna, Madam, Principal of our College of Education of Telangana University, Dr. Jalandhar, delegates and participants of this program. A very good morning to all of you. On behalf of Telangana University and my own, I welcome you all. The topic of this international conference is very important in this COVID-19 pandemic crisis. It is the need of the hour, as the life sciences industry is very important in terms of human health, in understanding the nature of disease and curing patients. Personalized medicine through advancements in genetics and healthcare procedures, collaborative innovative technologies of various life sciences industries, advancement of drug research, and increased digitalized assessment, diagnosis, and treatment of the patients are few of the top emerging trends in the life sciences. Machine learning and artificial intelligence are among the most important topics of digital transformation and are key to the growth of every life science industry. Life science companies utilize new latest innovative technologies which will help patients and avoid worsening conditions. Industries should be ready for new directions on large scale, from research and development to safety. The life science industry has been in spotlight in 2020 as government officials and the public pin their hopes for economic recovery on the discovery of an effective vaccine for COVID-19. Life sciences continues to be a very active market for both private investment and initial public offerings. There is much hope for a therapy or vaccine that will put an end to the pandemic and the fallout that has crippled economies. We can look ahead to where the economy and the life sciences ecosystem are headed. Life sciences tools and services are benefiting from a strong demand for testing services and supplies, reagents, and other components to support ongoing trials, COVID-19 tests, and manufacture of vaccines. It is believed that this demand persists well into 2021 as a vaccine is hopefully made available and testing continues within an increasingly open economy. The focus on COVID-19 therapies, critical medical devices, and testing capabilities has provided a halo effect for the life science industries. But bringing a vaccine to market, we believe life sciences will remain a primary focus for investment and innovation. I'm sure that this virtual mode international conference will be an opportunity for all the participants to discuss about emerging trends and novel techniques in life sciences. Deliberations, discussions, and exchange of views would be constructive, useful, and motivating. I hope it will render fruitful results to our healthcare system in this COVID-19 pandemic situation. I wish this event a grand success. I wish you all a happy, healthy, peaceful, and successful long life. Thank you. Thank you for giving me this opportunity.
Thank you, ma'am. Thank you. Now I request Professor Vidyavardhini madam to speak to introduce our opening speaker, who is giving opening address, Dr. Mahatab Bamji madam. Over to you, Vidyavardhini madam.
Thank you, Halim Khan sir. It's a privilege to introduce Dr. S. Mahata Banji. She's an INSA emeritus scientist from Dangoria Charitable Trust, Hyderabad, India. And she is a director, grade scientist who's retired from National Institute of Nutrition, Hyderabad. Dr. Banji got a PhD in biochemistry from botany from Bombay University while working at the Indian Institute of Science, Bangalore in the year 1961. After postdoctoral experience at the Tufts University of Boston and Johns Hopkins University, Baltimore, she joined the National Institute of Nutrition, Hyderabad in the year 1965 and retired from this institute as a director grade scientist, head of the biochemistry division and in charge of National Center for Laboratory Animals in the year 1994. She had made significant contributions in the area of nutritional biochemistry, including development of biochemical tests for early detection of B vitamin deficiency, deriving RDA from vitamins B1 and B2 for Indians, finding biochemical basis of skin lesions of vitamin B deficiency and lysine-carnitine relationship in animals and men. Since her retirement, Dr. Bamji has joined NGO Dangoria Charitable Trust DCT in Hyderabad and is currently Indian National Science Academy INSA Emeritus Scientist. She is trying to develop strategies for health, food, nutrition, environment, and livelihood security in villages of Medak district. The major focus is empowerment of women and adults and kids through social, scientific, and technological engineering and training in farm and non-farm skills. She has set up an award-winning rural food processing cum training center with guidance from CFTR in Mysore. Dangoria Charitable Trust received the National Award for Women's Development through Science and Technology from DST on March 8, 2013. Dr. Bamji has guided many students for PhD. She has published 100 full papers in peer-reviewed journals, written many review articles, book chapters, popular articles, and has edited a very popular multi-author textbook of human nutrition, now in its 4th edition. She is a fellow of Indian National Science Academy, National Academy of Agricultural Sciences, National Academy of Medical Sciences, and honorary fellow of National Academy of Science India, NASI, and AP and TS Academies of Sciences. She has received over 20 national and international awards. Dr. Bamji has been a member of many policy-making and advisory committees, including Vision 2020, Health Committee of TIFAC as co-chair, Planning Commission, Steering Committee on Science and Technology for the 11th 5-year plan, and chaired the National Task Force for Women in Science under the Ministry of Science and Technology from December 2005 to June 2007. She was Vice President of the Indian National Science Academy in charge of Science and Society program from 2009 to 2011. She is one of the honorary presidents of Jana Vijnana Vedika, People Science Movement, and takes keen interest in popularization of science. And she has received many awards like the Shakuntala Devi Amit Chand Award Prize of ICMR in the year 1971, Patwardhan Prize from ICMR in 1973, WHO Visiting Scientist Fellowship from Jan to March 1973, National Science Foundation Travel Award, USA 1983, Amrit Modi Award 1985, Dr. B.C. Guha Lecture Award of National Science Academy in the year 1987, Federation of Parsi Zoroastrian Association of India in 1987, B.C. Guha Lecture Award, Indian Science Caucus in 1997, Jawaharlal Nehru Birth Centenary Lecture, Indian National Science Academy 1998, Sri Kantiya Memorial Oration, Nutrition Society of India in the year 1999, National Women Bioscientist Award in the year 2000 from the Department of Biotechnology, GOI, Honorary Fellow of AP Academy of Sciences in 2002, Rustum Ramji Rotary Lecture Award in 2004, Distinguished Women Scientist Honor Academy for Science and Technology Communication, Hyderabad in the year 2004, the Chandrasekhara Venkataramana Medal of the Indian National Science Academy in 2005, Kamalapuri Sabarwal Lecture Award in 2008, Distinguished Alumni Award of the Institute of the Science Bangalore, IISc Bangalore, that is on centenary celebrations in 2008, Dr. Rajamal Devdas Memorial Award, Nutrition Society of India in November 2010, Achievers in Agricultural Hall of Farmer and Rice Foundation, Hyderabad in 2012, Professor Archana Sharma Memorial Lecture Award in 2012 by the Indian National Academy of Sciences, India, Indian Dietetics Association Founder Lecture Award in 2013, KT Acharya Memorial Lecture Award from the CSIR ICT in collaboration with the Oil Technologies Association of India in the year 2015, Living Legend Award of the International Union of Nutrition Scientists in the year 2017, Life Achievement Award of International Union of Food Science and Technology in the year 2018, C.R. Ramchandran Oration Award of Nutrition Foundation India in the year 2018. Thus, ma'am has vast experience in all the aspects of nutrition, and she has, it gives us immense pleasure to have madam. And thank you, ma'am, for giving me the opportunity for us, for the Department of Botany, to be over here. And over to Haleem Khan. Thank you.
Or to Bamji ma'am. Good morning, ma'am. Ma'am, unmute yourself.
Right. Can you hear me now?
Yes, yes, yes.
Okay, right, right. Thank you very much for inviting me and for the elaborate introduction. It's always a pleasure to speak to young scientists. At the age of 86, I enjoy my interactions with youngsters. Now, I'm sorry my video is not working, but since you can hear me, it's fine. Life sciences has many areas. Conventional sciences like botany, zoology, biochemistry, which came later, agricultural sciences, the more recent ones being cellular and molecular biology, biotechnology, immunology, and so on. Unfortunately, in the enthusiasm of more modern science, which is of course very important, some of the conventional sciences like taxonomy. I don't know how many of you in botany department are good taxonomists. I recall my dear friend, late Dr. Mohan Ram. If you went out with him in a forest or in a park, he could just pick out any tree, any plant, and give its name, its background, and so on. Today, we don't have too many taxonomists. One of the biggest breakthroughs in biological sciences took place with the discovery of vitamins in early 19th century. And this gave way to treatment of some of the raging diseases of the time, like beriberi, pellagra, scurvy, night blindness, blindness due to vitamin A deficiency, and so on. This was along with that, enzymes were discovered, and the whole science of biochemistry took birth, where we old-time bioscientists did not learn molecular biology. We learned only biochemistry. Unfortunately, many of today's molecular biologists, cellular biologists, don't have much knowledge of metabolism, but they have knowledge of other things. Uh, then came the discovery of the DNA, RNA, the genetic code, and so on by Watson and Crick. But let me tell you that though Watson and Crick got the prize for this, the credit should go to Rosalind Franklin, a woman who was sick at that time and whose X-ray pictures told, gave the information about double helix. Very often women are overlooked when it comes to recognition. Today, then for a long time, we associated nutrients like vitamins and minerals with certain diseases. But today we know that beyond these conventional diseases which are associated with nutrients, these nutrients, vitamins, minerals, etc., have more functions which we are not able to understand still. For instance, we know that vitamin D, which is the sun vitamin, it is generated in the skin through exposure to sunlight, is required for absorption of calcium and phosphorus and calcification of bones and teeth. But in addition, vitamin D also stimulates immunity. In fact, presently, with COVID raging, corona infection raging, there's a lot of interest in vitamin D, and vitamin D is being prescribed for fighting corona infection. So, and also today's research trend, biochemists are now also turning to cellular and molecular biology to understand how some of these, particularly fat-soluble vitamins, vitamin A, D, E, K, work at cellular and molecular level. Then Gregory Mendel is the father of Mendelian genetics, and that has revolutionized plant agriculture. Today, plants are being modified, tweaked to make them more nutrient dense, more environment friendly, etc. The classical example is the yellow rice, rich in beta carotene. Now, this is being done through conventional breeding or molecular breeding or biotechnology, genetic engineering. Now, for many people, unfortunately, misinformed, genetic engineering is a dirty word. They are propagating misinformation about how dangerous genetic engineering is. But this, of course, one has to have proper checks for safety, safety to health, safety to environment, and so on. But with this, this, thanks to the activities of some activists, there are hindrances to this, the modification of plants, particularly through genetic engineering progressing in this country. And a lot of enthusiasm is now being diminished. Then we know today that apart from the genetics, apart from DNA, genetic information, phenotype can be expressed without altering the gene sequence, without altering the nucleotide sequence. And this is called epigenetic effect. And this epigenetic effect is brought about through DNA methylation, histone modification. And this is brought about through nutrients, vitamins, minerals, and also environmental factors. Some of them will have good effects, some of them may be harmful. So it's not as if your phenotype is just dependent only on your genes. There are factors which go beyond genes, epigenetic effects, which determine your phenotype. Also, We are controlled by the bacteria, microbiome in our body. There are millions of microbiomes, particularly in the gut. And the science of prebiotics and probiotics and prebiotics has assumed new significance. Probiotics are the good bacteria which promote health. And in our diet, there are fibers which are not digested by human beings, but the good bacteria use them as food, and they determine our health. Then there are substances in plants, particularly plant foods, uh, pulses, nuts, certain green leafy vegetables, etc., which have what are known as prebiotics. And these prebiotics promote probiotics. So vegetarian food is very, very important for health. I'm not against non-vegetarian food, particularly eggs. I feel eggs are very, very nutritious and they should be promoted. But uncontrolled dependence on animal foods, like happens in the West and now in China also, is destroying the environment because the food conversion ratio, animals require more food to give 1 kg of meat. So while we in India should not say we should all become vegetarian, we should definitely promote eggs because very often eggs are not even fertilized. And it's a waste product for the bird. But at the same time, depending too much on animal products will have an harmful effect because you will need that much more fruit, which is not advisable in the present context. Then there are what are known as functional foods. This is another new science. Functional foods are are not nutrients, known nutrients, but there are some unknown chemicals, biochemicals, again present, phytochemicals present in plants. And these functional foods, today we have to recognize them and try to find out what is the extent, or what is their requirement. This is completely a new science where nothing is being done so far, though we do know functional foods. Then today, uh, even the known nutrients, how much is enough? How much should a person eat? This is called recommended dietary allowance. It's an old-fashioned science, but it has to be revised every year. Just recently, this year, just a couple of weeks back, the health minister released the new copy of recommended dietary allowance from National Institute of Nutrition. How much is enough? Unfortunately, today, India and many other countries are facing what is known as the double burden of disease. On the one hand, the old-fashioned diseases, pre-transition diseases, infections, and undernutrition, malnutrition are persisting. Today, we are facing the problem of obesity. Just a few days back, the most recent National Family Health Survey 5 report has come out. And that shows that almost 40% of people in Telangana and other places also are suffering from overweight and obesity. And at the same time, there is a vast population of women and children that is still undernourished. Anemia is a raging problem. We call the micronutrient deficiency. Micronutrient means vitamins and minerals. These deficiencies are referred to as hidden hunger. If a woman is anemic, you won't recognize her. She will go about doing more work than you and I do. But she is not adapting. She is compromising and she'll break down any moment. So I was just reading that NFHS report. It has come in newspapers also. Times of India has, that 40% of our population, the women and children, suffer from anemia. Stunting you can make out. If a person is thin, you can say, oh, this person is malnourished. but anemia effect is hidden. And anemia leads to so many problems. The whole performance, how can a nation which is malnourished become productive nation? Today, nutrition is not receiving the extent of importance that it should receive. We have fortunately succeeded in bringing down the burden of communicable diseases, but diseases, the malnutrition still persists. And it is disturbing to find that between the NFHS 4 report and 5 report, malnutrition change has not been a reduction in malnutrition. It has happened. But of course, India is a vast country, and there are what are known as Bimaru states, Bihar, Madhya Pradesh, Rajasthan, Uttar Pradesh, Chhattisgarh, and all that, and even Orissa, where malnutrition is more. So you cannot develop, no nation can develop without the fundamentals: health, nutrition, education, and equity, particularly gender equity. We have advanced a great deal in nuclear science, in space science, and so on. But India is not progressing because it has not given sufficient attention to these basics: malnutrition, infection, illiteracy. Change is occurring, but not to the extent that it should. Now, previously we used to think that nutrition deficiency results in impairment of physical and mental performance, which it does. We in nutrition, we call the first 1,000 days as the most important. About 1,000 days from the time of conception to 2 years of days. This is the time when the pregnant woman has to be sufficiently nutritionally good, the newborn infants up to 2 years, because this is a captive period in which you can make some changes. After that, it is like trying to catch a horse after it has left the stable. And after that, if you keep on pumping food, you may have unnecessarily healthy linear growth. Another aspect which is not being understood properly is what is known as fetal origins of adult diseases. One second. I'm sorry, I can't talk to you now. Fetal origins of adult diseases, which says that intrauterine malnutrition changes the genetic composition in such a way that those who are born with low birth weight, in later life, if there is a change of lifestyle, they are more prone to what are known as non-communicable diseases, the degenerative diseases of age: diabetes, cardiovascular diseases, cancer, and so on. In fact, India is referred to as the diabetic capital of the world. So malnutrition not only affects physical and mental performance and productivity, but it also impairs and makes you susceptible. Now today, the degenerative diseases, non-communicable diseases, are receiving greater attention than in the past. They have become number one problem. So sufficient to say, now there are two terms which are regularly used: food security versus nutrition security. And they are often mixed, interchangeably used, but they are two different things. Food security implies each and every individual receiving balanced diet required for age, sex, physiological status, etc. Nutrition security includes food security, but it goes beyond food security. And for nutrition security, you have to have healthy environment. You can't expect to live in a dirty, infected environment and expect to be nutritionally secure, because whatever you eat with from the mouth will go the other way. So for nutrition security, it's not enough to have just food security. For nutrition security, you have to have healthy environment, clean food, sanitation, and so on. And this is one area where in recent years, thanks to Prime Minister's Swachh Bharat campaign, today use of latrines has gone up. And we are hoping that some improvement in the environment is taking place. This and this, if the same emphasis is put on balanced nutrition, it will make India a happier place. Now, having enough rice and wheat at national level is not food security. Food security requires that you have to have enough pulses. Today, pulses, oil seeds, we are importing. Though in last 2 years, my agriculture friends, scientists tell me that improvement has been made. India is the largest producer of milk. It is the second largest producer of fruits and vegetables. Despite this, if you look at the diets, they are very deficient in animal products as well as fruits and vegetables and pulses. 35% of food is wasted. And this is an area where India needs to do a lot more. In countries like Philippines, 90% of vegetables are processed. We don't have enough facilities we are allowing food to be wasted in field, wasted after production, and also wasted in plates. If you see the amount of food that is wasted during our marriage and other functions, it's phenomenal. So again, to repeat and emphasize, I think India will progress only. Today, Punjab is producing rice. It has got no business to produce rice. It is depleting the water, groundwater, and it should diversify. Food production has to diversify. Monoculture, just this rice wheat cycle is more harmful than helpful. So despite all this agitation that's going on, I feel that the agitators should have very careful discussion with the government and remove, maybe the present rules can be modified, but they have certain strengths. With these few words, I once again wish this conference great success. Thank you very much.
Thank you very much, ma'am, for elaborate happenings in the life sciences and till current update, how the science has taken its plateau. And now I request Dr. V. Jalandhar to begin the technical session 1 and introduce our today's speaker from United States of America, Dr. Shyam Kumar Bhandari. Over to you, Jalandhar. Dr. Jalandhar, over to you.
Very good morning to all. Myself, Dr. V. Jalandhar, faculty in the Department of...
Jalandhar, your sound is very low.
Okay, sir. I will check. Hello?
Your sound is very low, Dr. Jalandhar.
Sir?
Okay, you continue.
Audible, sir?
You are audible, but your sound is very low. Okay, because Dr. Shyam is waiting since 50 minutes. So you introduce him quick.
Okay, sir. Okay, sir. I'm audible, sir?
You are audible. Okay, you start.
Okay, sir. Very good morning to all. It's my immense pleasure to introduce Dr. Shyam Bhandari Kumar. He is working as Senior Scientist, Department of Pathology, University of Alabama at Birmingham, USA. He has completed his school education and intermediate and graduation from Mother State, Andhra Pradesh, India. He has completed his post-graduation from Bangalore University, Bangalore, Karnataka State, India during 2006-2008. Then he has joined as a JRF at CCMB Hyderabad, India 2009 to 2011. He did his PhD from University of Münster, Germany. He has completed his PDF from Department of Pathology, University of Alabama at Birmingham, USA. Then he has felicitated by many awards, such as Best Student Award in PG program from Bangalore University, Bangalore in 2008. Then he has received gold medal, attaining highest merit in life sciences from Usmania University, Hyderabad, India. Then he has received merit scholarship from Government of India under graduate fellowship, Andhra Pradesh Government, India. Then travel award from American Society for Matrix Biology in 2018. He has completed research posters and oral presentation and published 60 research articles in international and national journals. And his research area is his expert in cancer biology, glycobiology,
Dr. Jalandhar, your audio is problem. You're not audible.
Hello, I'm audible, sir?
Yes, yes.
Okay, sir. He is expert in cancer biology, glycobiology, developmental biology, and drug delivery using exosomes, cancer diagnostic markers. Thank you, sir. Thank you for giving this opportunity. Over to Shyam Bhandari, sir.
Dr. Sham, welcome and good morning. I don't know what is in America. Good
So
morning.
it's almost, I would say it's 11:23 p.m. So it's night.
Oh. Okay.
Yeah. So
Start your presentation.
yeah. So shall I start?
Yes, you can start. You start your
So,
screen
so
sharing.
I started the sharing my screen. So I don't know whether it's visible or not. Okay. So yeah. So good morning, everyone. Thank you very much, Dr. Jalandhar, for kind introduction. And I would like to thank Dr. Haleem Khan, sir, and all the dignitaries for giving me the opportunities to share my research. So first of all, this talk is very kind of special for me because I did my bachelor's from Giriraj Government College, Islamabad, and that's where I started my biotechnology career. So that's why I think this talk is very special for me. So today I'm going to talk about the small extracellular vesicles, the next big thing. So to begin with, yeah, so these are the top contents. So initially I'll talk about the introduction to the extracellular vesicles and then followed by biogenesis of extracellular vesicles. Then I'll talk about what is the cargo that is present in the extracellular vesicles and what are the functions of extracellular vesicles. Then I'll talk about the initial experiments that was done using the mouse reticulocytes, which paved the way for the discovery of extracellular vesicles. then followed by, I'll talk about the isolation, purification, characterization of extracellular vesicles. Then I'll talk about the extracellular vesicle research in plants. So this is, I think, almost after 15 years, I'll be talking about plants. So, and then followed by a therapeutic application of extracellular vesicles, then followed by a biology of extracellular vesicles in COVID-19. Then at the end, I'll talk about my research on extracellular vesicles in cancer. And at the end of the talk, I hope I'll be able to convince you guys that indeed extracellular vesicles are the next big thing. So what are extracellular vesicles? Extracellular vesicles are heterogeneous group of lipid bilayer vesicles, which are secreted from almost all the cells in our body, in the animals, and from the plants. So depending upon the size of the extracellular vesicles, they are basically classified into exosomes, microvesicles, and apoptotic bodies. Exosomes are basically 30 to 100 nanometer in diameter. As you can see here, these are 30 to 100 nanometer in diameter, and microvesicles are 100 nanometer to 1 micrometer, and apoptotic bodies are more than 1 micrometer. So as you can see here, this is the size of the cell, so which is approximately in between 8 to 12 micrometer, and apoptotic bodies are almost equal to the platelet size, and microvesicles are almost equal to the size of the bacteria. Exosomes are equal to the size of the virus. So we are dealing with the very, very tiny, minute particles. So these are extracellular vesicles. So how these extracellular vesicles are secreted from the plant. So what is the biogenesis of extracellular vesicles? So if you look at the biogenesis of extracellular vesicles, so first we talk about the microvesicles. So these are formed by the outward budding and fission at the plasma membrane. And exosomes are formed by the inward budding of endosomal membrane, and these exosomes are released into the extracellular space. This exosome biogenesis basically is of two types. So one is S-cut dependent and the other is S-cut independent. So this S-cut dependent mechanism involves the proteoglycan syntenin protein, and Alix complex. And S-cut independent mechanism involves the tetraspanin proteins and lipids such as ceramide. So this is the biogenesis of extracellular vesicle. So what are the contents in the extracellular vesicle? So what exactly are present inside the extracellular vesicle? So if you look at the contents in the extracellular vesicles, so this is the extracellular vesicle. So this is the magnified image of single extracellular vesicle. So as you can see here, this is a lipid bilayer membrane, and so it has a lipid bilayer membrane, and it consists of tetraspanin proteins, and it also consists of the cholesterol. It is also made up of the cholesterol. And if you look inside the extracellular vesicles, it has microRNAs, long non-coding RNAs, other non-coding RNAs, lipids, proteins, and mRNAs and DNA. So basically, these are packed with all biologically active molecules. So this is the content of the extracellular vesicles. Then, so what is the exact function of these extracellular vesicles? So basically, if you see, this is the secreting cells where it is secreting the exosomes. So what cell does is basically it packs all the information into these extracellular vesicles, and it delivers these extracellular vesicles into the extracellular space. Basically, these extracellular vesicles contains the signature molecules from the secreting cell. And once they are released, so these are taken up by the recipient cell. So it is going and fusing with the plasma membrane of the recipient cell. So once it fuses with the plasma membrane, it basically diffuses all its contents, basically it delivers all its contents, such as DNA, protein, microRNAs, lipids, everything into the recipient cells. Once those biologically active molecules are delivered into the recipient cells, that will stimulate the cell signaling that alters the gene expression of the recipient cells. And it also suppresses or activate the immune system. So this is the basic function of extracellular vesicles. So till now, what we have learned is what are extracellular vesicles and what is the function of extracellular vesicles and what exactly is present in the extracellular vesicles. So then I'll give you an experiment which paved the way for the discovery of extracellular vesicles. So extracellular vesicles were first discovered in 1983, where the back-to-back papers were published in Journal of Biological Chemistry and Cell Journal. So these experiments were first published in these journals. So what exactly these people have done is basically C.V. Harding was the guy who discovered these extracellular vesicles. So he was a PhD student in Washington State University when he discovered these extracellular vesicles. So what he did was he was studying the transferrin receptor turnover in reticulocytes, in mouse reticulocytes. When the mouse reticulocytes matured, they lost the transferrin receptors. So he was studying exactly how is this trafficking of this transferrin receptors is occurring in mouse reticulocytes. So what he did was basically he took the mouse reticulocytes and then he incubated them with the colloidal gold conjugated transferrin protein. So when it was incubated, and then he did the transmission electron microscopy. So when he did the transmission electron microscopy, as you can see here, basically he found all the transferrin gold conjugated colloidal particles embedded in the vesicular body, and which is fused to the plasma membrane. So it was not diffused elsewhere, but it was only present explicitly in the multivesicular body. So that's when he identified these are multivesicular bodies and these are extracellular vesicles. So initially he got a doubt that it might be the artifact of like transmission electron microscopy, where you have to freeze the sections. So then he did the experiment like without freezing and with freezing. So in both the cases, he found the transferrin gold conjugated particles in the multivesicular bodies. So at the end, he came to the conclusions that, so these are extracellular vesicles, and this transferring is explicitly localized into these extracellular vesicles. Then the Rose Johnstein was the guy who gave the name extracellular vesicles. So this is the first experiment that was done in 1983, which paved the way for the extracellular vesicle research. So how do you isolate these extracellular vesicles? So extracellular vesicles can be isolated by different methods, but a commonly used method is differential centrifugation. So where it follows the centrifugation of the biological sample at the different speeds, at each speed, the different particles, such as at low speed at 300 G, the live cells will be sedimented, and then at 2000 G, cell debris will be sedimented, pelleted, and then at 10,000 G, the apoptotic bodies and microvesicles will be pelleted, and at 100,000 G, this is where you get the pellet of the exosomes with the contaminants, other contaminants such as the other associated proteins. Then finally, you have to resuspend this pellet in the PBS to get the centrifuge at 100,000 G one more time to get the pure pellet of exosomes. But still, this pellet is not that pure. Still, it might have some kind of a contaminants. So directly, we cannot use these extracellular vesicles for our research purpose. So, and the other thing that is available is basically the commercially available kits. So many companies such as Qiagen, R&D, and Fisher Scientific. So basically everyone came up with the kit for the isolation of extracellular vesicles. So what they use is basically they'll use the different chemical such as polyethylene glycol, sodium acetate, protamine, heparin based columns. So they use the different type of the chemicals to precipitate the extracellular vesicles. But the extracellular vesicles that you get from the commercially available kits are basically impure. They are associated with lots and lots of junk, so which you cannot use for your research purposes. So that's why the majority of the people use the differential centrifugation as the first step in isolating the extracellular vesicles. Then to further purify these extracellular vesicles, majority of the researchers use the density gradient centrifugation, that is sucrose or iodoxanol density gradient centrifugation. So what we do here is basically we use the 4 ml sucrose. So as you know, basically density gradient centrifugation separates the vesicles depending upon the density and the size and volume. So as you can see here, so when you load first you have to load the sucrose, and on top of it you have to load the 25 ml of extracellular vesicles suspended in PBS, and then followed by you have to spin at 1000 G for 75 minutes, and then remove the sucrose cushion. That's where your extracellular vesicles are there, and then spin and then dilute it with the 60 ml of PBS, and then spin it 100,000 G for 75 minutes. So this is where you get the clean pellet of exosomes that is devoid of all junk. So basically you can use these extracellular vesicles for all your in vitro and in vivo experiments. So where you can inject these pure exosomes into the mouse also. And you can also inject these exosomes into the humans also to study the effects of these extracellular vesicles. So they are that pure, almost like gold standard pure. So then there's one more kind of purification method where people use the tetraspanin proteins which are present on the surface of the extracellular vesicles. So they will be conjugated like the CD63, CD9. So these are basically present on the surface of extracellular vesicles. These are the proteins present on the surface of the extracellular vesicles. So these, basically the gold particles or magnetic beads will be conjugated with these proteins, and the immunoprecipitation will be performed where you can get the pure extracellular vesicles. That is also one of the method where you can get the extracellular vesicles in a pure form. Then followed by size exclusion chromatography, and then now the people are using microfluidic devices in order to purify the extracellular vesicle. These microfluidic devices, what you have to do is basically if you want to isolate the extracellular vesicles from the urine or from the blood or from the serum or from any biological liquids, so you have to pass it through the microfluidic devices. So these have a kind of a pores where it will trap the extracellular vesicles, and that's where you can purify these extracellular vesicles using the microfluidic devices. And how do you characterize these extracellular vesicles? So once you isolate the extracellular vesicles, so you wanted to know whether the things that you have isolated are extracellular vesicles or some kind of a debris. So basically, the researchers use transmission electron microscopy to visualize these extracellular vesicles. So as you can see here, this transmission electron microscopy uses the beam of electrons to produce a magnified image of a specimen or a sample. So this is the transmission electron microscopy image of a single extracellular vesicle. So these are the single extracellular vesicles that are, this image was taken at the facility in our lab. So this you can see here, this is how the extracellular vesicles look under transmission electron microscopy. So then if you wanted to count the number of extracellular vesicles that are present in your isolated solution, so there should be an instrument, right? So this is the instrument which basically measures the number and size of extracellular vesicles in the solution. So this is a nanosite. This instrument was developed by Cambridge. As you can see here, basically it has so this is the machine actually. So it uses the laser, and it has a microscope attached from top of it. So what it does is basically you have to inject the sample from here. So as you can see this syringe here, so you can inject the sample from here. So it will go inside, and then this is the apparatus where it is exposed to the laser. And once this is exposed to the laser, the particles in the solution will get scattered the light, and the light will be captured by a microscope, and that microscope will give kind of sense and information, and where you can see this is a graphical user interface where you will get the size and number of particles. As you can see here, this is how the extracellular vesicle looks when you see in the on the computer. So this is each single dot is the extracellular vesicles. So the brightest dot is the larger size, and the kind of the one with the lower intensity of light is basically of a smaller size. So, and this is how the peak you get after processing this data. So as you can see here, this single sharp peak, which is approximately around like average mean of 95 nanometers. So this is how the extracellular vesicles or exosomes are quantified using the nanosite. Then, so, and if you wanted to further characterize these exosomes, so you can characterize these exosomes by the presence of the different proteins on its surface. As you can see here, so there are different proteins which can be detected in the exosomes. As you can see here, this is the flotillin, clathrin, CD63. These are present in the exosomes. They are absent in the cells. So as you can see here, this is the Western blot performed on the cells and as well as the exosomes. So this flotillin, clathrin, CD63 are enriched in the exosomes. So this is how you detect the exosomes. So till now, what we have learned is basically how we characterize the exosomes using the nanosite and then Western blot analysis. Then further, so I would like to switch into the what's the role of these extracellular vesicles in plant research. So as you can see here, so Rogers was a guy who performed an experiment. So he was studying basically the cell death. So he wants to study how the cells die and how this leaf will die. So what he did was he took the Arabidopsis thaliana plant and infected it with the bacteria. And after infecting it with the bacteria, so he cut the Arabidopsis thaliana leaf into the slices. He made the sections of it, and he observed it under the transmission electron microscopy. So this is the image of a transmission electron microscopy, as you can see here. So there are small vesicles that are present between the cell wall and plasma membrane. So this he called as extracellular vesicles. So this is the, exactly, so what he did was he wanted to know what is the role of these extracellular vesicles in plant kind of biology. So what he did was he purified these extracellular vesicles and he did the mass spectroscopy analysis. So mass spectroscopy analysis, where you can detect the different proteins present in the extracellular vesicles. When he did the mass spectroscopic analysis, he came to the conclusion that these extracellular vesicles that are secreted from the Arabidopsis thaliana plants after infecting with the with a bacteria, so they have the cargo of they have the cargo of the proteins that are related to the plant immunity. So basically, the plant is fighting the pathogen by packing the biologically active molecules inside the extracellular vesicles. So then he came to the conclusion that these extracellular vesicles has the biologically active molecules. They can be helpful in cell wall building enzymes involved in plant growth and development. And it also plays an important role in plant microbe symbiosis, where the plant also releases the extracellular vesicles, so that will communicate with the microbe. And in the other way, the microbe also releases the extracellular vesicles that will communicate with the plants. So, and then they are also involved in the pathogen defense protein mechanism. So where this plant will kind of release all the biologically active components to fight the pathogen. And then it also has the small interfering RNAs that can silence the pathogenic genes in the pathogen. So I'll give you a best example. So in order to see how exactly what happens when a kind of a fungi infects the Arabidopsis thaliana plants, how the plant fights that fungi by using the extracellular vesicles. So you can see here, basically, so the Sia et al., they have published a paper in Science in 2012. So what they did was basically they used the same Arabidopsis thaliana plant, and they infected that with the fungal pathogen Botrytis cinerea. And when they infected that, and then they took the leaf and they did the slice of this section of the leaves, and then observed it under the electron microscopy. As you can see here, exactly at the intersection of this fungal pathogen and Arabidopsis thaliana cells. So you can see extracellular vesicles that are getting released from the Arabidopsis cell. So as you can see here, clearly this cell is Arabidopsis thaliana cell is releasing the extracellular vesicles in order to fight this pathogen. And what they have also found was, and then what they wanted to know was basically, okay, so this confirms that these extracellular vesicles are released into the extracellular space, right? Then whether these extracellular vesicles are taken up by this fungal pathogen or not. So they wanted to know that. So then they did the in vitro experiment where they took the fungal pathogen and infected that with the GFP labeled exosomes. So basically you can label the exosomes by using the different dyes. Basically this has a lipid, right? It has a lipid bilayer. So you can label them with the lipid dyes like as PKH26 or DIL. So there are different dyes which are with which you can label these extracellular vesicles. So what they did here was, so basically they labeled the extracellular vesicles with the GFP protein, and then they added to the fungal pathogen in absence of Triton X. So Triton X is a detergent, and so these are lipid vesicles. So when you incubate the extracellular vesicles with the Triton X, so if they are outside, they'll get scattered. So they'll lyse, right? And after 2 hours, so as you can see here, so this is a control. So where you can see here, there's nothing is changed. But when you add the extracellular vesicles and incubate for 2 hours, in the absence of Triton X, so you can see here all the extracellular vesicles are inside the pathogen, inside the fungal pathogen. And in presence of the Triton X also, they are still present inside the fungal pathogen. So which means that, so these extracellular vesicles are taken up by the fungal pathogen. So they are internalized by the fungal pathogen. So then further what they did was they extracted the they extracted the proteins and they extracted the small RNAs from this fungal pathogen. And what they found was, so they found the Arabidopsis thaliana sRNAs, small RNAs and proteins in the extract of this fungal pathogen. So which means that indeed these extracellular vesicles that are produced from the Arabidopsis thaliana are taken up by this pathogen, and the contents are getting delivered to this pathogen. So then further what they did was they studied what's the impact of this small RNAs that are delivered to this fungal pathogen. So what they found was this small RNAs are basically trying to reduce the pathogenicity. They are trying to reduce the genes that are genes responsible for the fungal pathogenicity. So they confirmed it by using the mass spectroscopic analysis. So this gives a clear indication that the plant cells are communicating with the fungal pathogen. They are kind of fighting the fungal pathogens by extracellular vesicles, by biologically active components in the extracellular vesicles. And further, so what are the applications of this therapeutic applications of this extracellular vesicles? So till now what we talked about was I gave the example of one example of extracellular vesicle research in plants. There are plenty of examples where people have studied the different role of extracellular vesicles in plant defense mechanism and different other biological prospects. So this extracellular vesicles are best suited for therapeutic applications. So these are not only involved in basic biogenesis of this different biological process, but they can be used as delivery vehicles. So you can ask me a question, why? So why do you think that this can be used as an kind of a delivery vehicles? One of the major quality of the delivery vehicles, as you might have known, so the liposomes are used as a drug delivery vehicles, right? Or some kind of a nano vesicles can be used as a drug delivery vehicles, right? So in the similar way, these are small particles. So as I have told, so these are almost kind of similar to the size of the viruses. So, and these are lipid vesicles, and these are very stable. So as I said earlier, these extracellular vesicles are present in all biological fluids, which means that they are very stable at various different pH conditions. And in presence of different enzymes and different kind of situations, they are very much stable. So that's why they can be used as a delivery vehicles. And these extracellular vesicles are taken up, these are secreted from almost all the cells in our body and are also taken up by all the cells in our body. So this makes them the ideal vesicles for delivering some you can pack the different drugs, or you can pack the nutraceuticals, or you can pack the anything you want, like therapeutic protein, or you can pack the DNA, RNA, or small interfering RNA into these vesicles, and you can use it as a delivery vehicle. And as I said earlier, so basically these are present in the breast milk. So this is also important thing. So the scientists have also studied the extracellular vesicles in the breast milk. So what they found was the breast milk contains the lot of extracellular vesicles packed with the different proteins, DNA, mRNA, which are beneficial for the baby growth. So that's what they found. So these are very, so these extracellular vesicles that are present in the breast milks were important for the baby growth. And one more interesting thing was, so whatever we eat, so we eat a lot of fruits, vegetables, so what you name it actually. So everything has extracellular vesicles in it. People have isolated the extracellular vesicles from citrus plant, lemon. They have isolated the extracellular vesicles from broccoli. So they have isolated extracellular vesicles from cabbage. So you name it actually. They have isolated the extracellular vesicle from those sources. And as I said earlier, so this can be used for kind of loading different cargo, such as protein, DNA, RNA, and nutraceuticals. So there are two different ways of loading the cargo into the extracellular vesicles. So one is passive cargo loading, and the other one is active cargo loading. So passive cargo loading is where, so you incubate the extracellular vesicles with the thing which you wanted to pack inside the extracellular vesicles. Let's say some kind of a protein. So you incubate together. So what happens is basically with the process of diffusion, this protein will enter into the vesicle and get encapsulated into that extracellular vesicle. And in active cargo loading, so what you do is basically you will lyse these extracellular vesicles and then incubate that. You can kind of load the kind of lyse these extracellular vesicles and incubate them with the different components such as different drugs, proteins, and DNA and RNA. So, and then by itself, they again form the lipid bilayer vesicle. So they kind of rearrange themselves. So after lysing also, they can form kind of a same extracellular vesicles, and the protein gets packed into those extracellular vesicles. So this is the active cargo loading. So these are two different methods by which you can pack the extracellular vesicles with the desired protein, DNA, or nutraceutical or drug of your interest. So then the question comes is basically, so for the kind of a drug delivery things, you need the vesicles in a liter quantities. Like you need a huge number of extracellular vesicles, right, for the kind of a delivery when you use it for the delivery. So what you can do basically is, so you can culture these extracellular vesicles in the bioreactors. You can kind of churn, you can make the plants to secrete a lot of extracellular vesicles, and you can purify those extracellular vesicles using the bioreactor. You can grow them in the bioreactors, and you can make tons of extracellular vesicles. So biogenesis of it is not at all a problem. So this is also one of the interesting feature that makes them kind of ideal for usage in the therapeutic application. So I'll give you a best example. So let's say, so this is the one scientist, one crazy scientist. What he did was basically he wanted to know whether the lemon-derived extracellular vesicles can be used for treating the gastric cancer. So what he did was, so this is the first figure. So he isolated the extracellular vesicles from the lemon. And as you can see here, this is a TEM image showing that each extracellular vesicle. So as you can see here, and then what he did was he basically did an in vitro experiment first. So he took the gastric cancer cells and added these extracellular vesicles, which are labeled with the DIL. So as I mentioned earlier, DIL is basically a lipophilic dye, a red colored dye, which get integrated into the extracellular vesicles. So this is a red. So red specks, what you are seeing here, these are all extracellular vesicles. And this is the Haw staining. Basically, it stains the DNA in the cell. So basically, this is the cell. And when you merge these two together, so they are kind of co-localizing. So these extracellular vesicles are basically getting delivered to the gastric cancer cells. So this is a single cell. And they did this spheroid model. So this is a 3D system model. So they did the same thing. So here also they can see here. So this extracellular vesicles are getting delivered to the, this is a merge image where you can see the extracellular vesicles are getting delivered to the cells. So with this, they have confirmed that this lemon extracellular vesicles can be delivered to the gastric cancer cells. So further, so now they wanted to know, okay, these extracellular vesicles are taken up by the gastric cancer cells in vitro. Then what about this in vivo? So to test this in vivo, what they did was they used a mouse model. So they took the Babsy mice. So and what they did was they injected the gastric cancer cells into the mice, and they have developed a tumor. As you can see here, this is the luminescent image where you can see this is this color, wherever the color is seen. So this is basically, so this is the gastric area, so where the tumor has developed because they have injected the gastric cancer cell. And then what they did was they injected the lemon derived exosomes into these mice, and first experiment they injected into that mice, and they let it go for almost like every 2 to 3 days. They injected 50 microgram of lemon derived exosomes, and each day they have measured the tumor size, and not each day but 3 to 4 days they have measured the size of the tumor. So first thing what they wanted to know was, so they wanted to know whether these extracellular vesicles that are derived from the lemon, right? So they have acidic pH, right? So they wanted to know whether that can destroy or that can change some of the organs, whether it change the morphology of the organs, whether anyway it impacts in a bad way to the mice. So they wanted to know. So what they did was, after the experiment was done, so they took the mice heart, liver, spleen, and lung, kidney, and they did the sections of it, and they did the HME staining. So what HME staining does is basically it stains the nucleus, and it also stains the hematoxylin basically stains the nucleus, and eosin, it stains the extracellular matrix and cytoplasm, as you can see it in pink. So basically with this staining, they have compared the without injecting the extracellular vesicles and with injecting extracellular vesicles, is there any change in the morphology of the different organs, cells in the different organs? So as you can see here, there is no change in the morphology of the cells in the different organs. So with this, they came to the conclusion that there is no negative impact of these extracellular vesicles on any morphology of different organs. So this is good. So this they kind of sorted out this problem. So they have cleared this problem. So then they measured the tumor size. So what happened to the tumor size after 19 days after treatment with the lemon extracellular vesicles? So as you can see here, so this is a tumor, so which is kind of isolated, and as you can see here, these are different days, different tumor sizes. And as you can see at the end of the experiment, so you can see the tumor kind of is still there in control mice as expected, because we didn't treat them with extracellular vesicles. But when you treat it with the extracellular vesicles, the tumor size is reduced, drastically reduced. You compare these two, so it's kind of a drastically reduced. So which indicates that this gastric cancer therapy worked. So lemon-derived extracellular vesicles kind of reduce the gastric tumor. So this is kind of one of the examples where the extracellular vesicles can be used in treatment of gastric cancer. There are many experiments, there are many researchers where they have shown these extracellular vesicles can be used for treating different types of cancers and different other diseases. And so now I'd like to focus the extracellular vesicles research in COVID-19. So I won't take much time. So I'll give just a kind of a brief things about the extracellular vesicle research that is going on in COVID-19 related viruses. So as you can see here, so what experiment they did was basically they isolated the extracellular. So as I said earlier, so almost all the organisms secrete the extracellular vesicles, be it the plants, fungi, virus, animals, humans, and everything. They secrete the extracellular vesicles. So in a way, this COVID-19 also secretes the extracellular vesicles. And what scientists have done, that they have isolated these extracellular vesicles from the COVID-19 virus, and they have infected that with the lung epithelial cells. As we know, the COVID-19 primary target is lung. So they wanted to know whether these extracellular vesicles that are derived from the COVID-19 can induce the lung fibrosis or not. So when they did the in vitro experiment, so what they found was these COVID-19 extracellular vesicles are indeed inducing the lung fibrosis. Then some more researchers, what they did was they wanted to know whether these extracellular vesicles can be used as a kind of detecting the disease. So if you wanted to detect the early disease, you can isolate the extracellular vesicles from the patient blood. Like they have did the multi-center study where they have collected the patient blood, and then they isolated the exosomes from those patient blood, and they analyzed the different proteins inside proteins and RNAs in the extracellular vesicles. And what they found was they have the antiviral response proteins, they have coagulation related proteins, and they have the liver damage related proteins. So which indicates that the person is infected with the COVID-19. So basically, so when the person gets infected with the COVID-19, the cells are under stress. So what cell does is basically it is trying to fight out the pathogen. So it packs all the information that is inside the cell and it dumps into the extracellular vesicles and it releases into the extracellular space. And this can be isolated, and you can see if more antiviral response proteins are there, so then you can see more infection. So in the same way, like if you see if there is an advanced stage of COVID-19 infection, so you see more kind of organ damage related proteins inside the extracellular vesicles. So this is how the extracellular vesicles can be used for COVID-19 early detection, early prediction detection. Then further, so what people have also done was, so they use the, as I have mentioned earlier, so this extracellular vesicles can be used for delivering drugs, and they can be used as delivery vehicles, so to treat different kind of diseases. So in the same way, as you can see here, so scientists, what they did was they isolated the extracellular vesicles from mesenchymal stem cells. So mesenchymal stem cells derived exosomes are packed with the different cytokines, so which can fight out this pathogen. So when scientists have incubated these extracellular vesicles with the SARS-CoV-19 in vitro, what they found was, so these extracellular vesicles are able to kind of reduce the replication of the virus, kind of reduce the replication of the virus. So basically, these extracellular vesicles are able to fight those those viruses. And one more approach was scientists tried to kind of encapsulate the different drugs that are used for treating the COVID-19. As you can see, remdesivir is one of the drugs, and people are using dexamethasone, and there are different drugs which are used for treating COVID-19, right? So they are trying to encapsulate those drugs in the extracellular vesicles, and they're trying to treat those kind of, they're using as a delivery vehicles to treat the COVID-19. And further, so they have also used they're also using these vesicles for kind of packing the vaccines in these extracellular vesicles. And in the same way, they're also using the platelet-derived extracellular vesicles to fight out this COVID-19 infection. So COVID-19 infection can be also kind of, scientists are trying to cure this kind of reduce this infection by using this mesenchymal stem cell derived exosomes, or with the drugs, packing the drugs inside the extracellular vesicles. And they're using different approaches for treating this COVID-19 by using the extracellular vesicles. So this sums up that the extracellular vesicles are present everywhere. So name it, virus, bacteria, plants, everywhere. So this is a kind of active research where lots of researchers are working on. So why I'm saying that this is the next big thing, because almost on an average, so there are almost 1,000 to 1,500 articles are getting published on extracellular vesicle each year. So each year, almost 1,500 articles are getting published on extracellular vesicles. And there is a lots of funding for extracellular vesicle research from National Institute of Health. And there is a one dedicated journal for publishing the extracellular vesicle related stuff, that is Journal of Extracellular Vesicles. And if you wanted to know whether your protein of interest is present in the extracellular vesicles or not, so there is a specific database called ExoCarta, so where it lists, so whoever kind of identifies any proteins in the extracellular vesicles, that is deposited into this ExoCarta website. So you can look for your protein of your interest in the ExoCarta. And as I have said earlier, so there are multiple extracellular vesicle based therapeutics are in advanced stages of clinical trials for cancer and other disease treatment here in US and elsewhere. So as you can see here, not only in the biology but also kind of they can be used as a delivery vehicles for treating the different kinds of the diseases. So at the end, so what I would like to do is I would like to kind of briefly tell what exact research what I am doing by using the extracellular vesicles in my current lab. So our lab works on a protein called heparinase. Basically, this heparinase is a protein or enzyme. It cleaves the heparin sulfate chains. And by cleaving that, what it does is basically it releases the different growth factors that is bound to this heparin sulfate chains. And our lab has established that if a myeloma patient express high levels of heparinase, so that is not good. So it enhances the tumor progression. So it alters the gene expression and it remodels the extracellular vesicles. The take-home message is basically if the patient express high levels of heparinase, that is bad for the patient. So that leads to the metastasis, that leads to the chemo resistance. So I'm working on that protein. And as you can see here, this is a PET scan imaging of a multiple myeloma patient. So these black dots, what you can see here are the lesions, multiple myeloma lesions. Basically, you can see it in all the long bones. So when you look at these lesions under the microscope, so you can see heparinase there. So which means that this heparinase is present in these lesions. So this is bad. I mean, this is kind of bad for the patient. So if the patient expresses high heparinase, then there's a high chance of tumor metastasis. So that's what we have established in our current lab. And further, what we have also shown was one of the interesting features. What is the link between the heparinase and extracellular vesicles? So we have published a paper in Journal of Biological Chemistry. This is a paper of the year in Journal of Biological Chemistry. So where we have shown that the heparinase enhances the myeloma-derived exosome secretion, composition, and function. So this shows that the heparinase has a role to play in exosome biogenesis or extracellular vesicle biogenesis. And further, what we have also shown was, so when you treat the patient with the chemotherapeutic drug, so this heparinase level get enhanced. So this is the baseline where in the 9 individual patients, we have checked for the levels of heparinase before the treatment. So you can see here there's a heparinase level is present. But when you increase the different treatment cycles, so this is the first stage of treatment. So you can see the heparinase is even more increased. And if you do the second stage, and it's even more increased. So which means that the chemotherapy is basically enhancing the heparinase level. So that's not good. So chemotherapy is not good. Chemotherapy is kind of trying to help the tumor by enhancing the level of heparinase. And so the question what I asked is basically if the chemotherapy is bad, so what is the effect of that chemotherapy on extracellular vesicle secretion? And how does this affect the chemotherapy on heparinase levels in the exosomes? And I wanted to know the chemotherapy derived extracellular vesicles role in tumor cell growth as well as tumor host interactions. So for that, I used the multiple myeloma cells, and I treated them with the different multiple myeloma drugs which are used for treating the multiple myeloma, such as bortezomib, carfilzomib, and melphalan. And then I isolated the exosomes and I quantified them using the nanosite, as you can see here. So when I don't treat the cells with the drug, so you don't see, you see exosomes, but it's not much. But when I treat with the bortezomib, so there's a huge increase in the extracellular vesicles. And same with the carfilzomib and melphalan. So basically the take home message is, if you treat the cells, myeloma cells, with the different drugs, so it is enhancing the extracellular vesicles biogenesis. And further, and when I looked for the protein of interest in these extracellular vesicles, I found that my protein of interest is enhanced in these extracellular vesicles after chemotherapy. So with increasing dose of chemotherapy, my protein of interest is getting enhanced. So that's bad. That's bad. So the chemotherapy is bad for treating multiple myeloma patients. So it's same is the case with the carfilzomib and melphalan. So, and when I did the proteomics analysis of those exosomes, and what I found was they're all kind of loaded with the cell cycle proteins, which helps in the tumor cell growth and proliferation and metastasis. So finally, what I found was, so when you treat the multiple myeloma cells with the chemotherapy, there is a huge burst of extracellular vesicle release, and these extracellular vesicles are taken up by the multiple myeloma cells, and as well as they enhance the different chemo resistance pathways, and they enhances the cell proliferation, and they also enhance, they are also taken up by the macrophages, and together this will promote the aggressive myeloma. So when you treat the patient with the chemotherapy, the tumor goes high. So that's what the chemotherapy doesn't work in many patients. This might be one of the reasons why, because the extracellular vesicles are getting secreted with the different cargo that helps in the tumor growth and chemo resistance. So these are my further works that I am continuing. So I am looking at the role of extracellular vesicles in chemo resistance in multiple myeloma, and I'm working on a role of extracellular vesicles in bone damage in multiple myeloma, and I'm looking at the role of extracellular vesicles in angiogenesis, and I'm also looking at the role of extracellular vesicles in exosomes in metastasis, tumor metastasis. And I would like to target these chemo exosomes. I would like to design a compound where I can stop this chemo exosomes uptake by the tumor cells. So I'm also working on different therapeutic approaches to inhibit the chemo exosomes or exosomes uptake by the tumor cells. So that's it. So thank you very much. Thank you very much for giving me the opportunity, and I'll be happy to take the questions if you have any.
Dr. Sham, pardon, very big thank you for accepting our invitation and giving such a beautiful, wonderful talk on your work, what you are carrying out. Now we are 1 hour delayed by the schedule. So kindly bear with us. We'll, if any question posted, we'll send you the email. Thank you very much. And now I request Dr. Devraj Srinivas to introduce our next speaker, Dr. Santosh Kumar. Over to Dr. Srinivas.
I would like to introduce our guest. It is my immense son, Dr. Santosh Kumar. Dr. Santosh Kumar sir is an eminent postdoctoral scientist at Faculty of Pharmaceutical Science and Chulalongkorn University, Thailand. He was awarded a PhD degree in the field of the plant biotechnology and obtained his PhD from Kevampu University, Shimoga. He was previously worked at the University of Agricultural Sciences, GKVK, Bangalore, and University of Guelph, Canada. His main focus was aimed to understanding the supply chain of herbal trade and authentication of herbal drug using genomic and the metabolomics approach. during his PhD, has analyzed the extent of alteration and substitution of highly traded herbal raw drugs products as a part of the international science and technology partnerships. ISTD, it is a project. He is a recipient of many awards such as the Rajkar Rachel Pisek Sampak won travel awards from Society of Economic Botany, SEP USA, International Barcode of Life, IBOL, Canada, and SHERP DST Goat of India. His research field, number of the scientific publications in international journals. He has current postdoctoral work involves the development of a standalone, the herbal product authentication database to provide use-friendly authentication approach for plant raw drugs. Sir, thank you for giving me this opportunity to introduce such a eminent personality. Thank you, one and all. Over to Speaker Dr. Santosh Kumar, sir.
Good afternoon, Santosh. Welcome to the international conference. Please, you can start your presentation.
Can you audible now?
Yeah, your audio and your video are clear.
Okay, thank you, sir, for a nice introduction, and thank you for Dr. Haleem Khan for invitation for this wonderful event, and also organizing committee and Department of Botany and Telangana University. So I'm going to start my today's talk. So you might have all know about recent event on herbal drugs, how the herbal drugs are most important in the present scenario of this COVID. And recently last 2 months 2 weeks back in a adulteration study reported in India, and that is related to honey. That news become a national and international news in the all over the world. So there's some group of the people from the Center for Science and Environment researchers. They took the branded honey samples from the different various companies, and they found adulteration 76%, nearly 76%, using the NMR metabolites. So this kind of things happening is not only for nowadays, since from the long times it will be happening. So our research is working on more than a decade. We working on herbal drug authentication service, authentication system for the safety and efficacy of the drug. So in this aspects, today I'm going to present our ongoing research and try to cover up all the whatever we done so far. So my title of this today's talk is Medicinal Plants and Trade, How Safe Are Herbal Drugs Now? So today these are my overview of my presentation. So I would just a quick overview of medicinal plants in trade, and what are the problems in medicinal plant trade, and how our study is addressing these questions, and what are the outcome of our projects. So you all know these plants having the long history, and plants have been used for the cure and heal throughout the human history and right now. And majority of the majority of the secondary metabolites we are depending upon plants, and plants having very extensively used so far now also. So if we come to Indian scenario, we believe in traditional medicines, and we have actually the four medicinal system: Ayurveda, Unani, homeopathy, and allopathy. But most of the medicinal plants coming from under the Ayurveda. So if you see the Ayurvedic product now in the market, they will come in the single form and also multi form of this. Multi formulations. Nowadays we'll get lot of different brands, and they will labeled as herbal or traditional, like that they will label and they will sell. So if you see the medicinal plants trade in India, see this is an old data. So 9,500 registered herbal industries are present. And if you see the graph, 40% of the in the medicinal plants are covered in Ayurveda. This Ayurveda is predominantly medicinal plants are covering, and more than 600 higher plants are used in herbal industries, and 960 medicinal plants are present available in the herbal. herbal industries. So if you see the medicinal plants turnover, this annual turnover, this is in 2014 to 2015, this data, nearly 5,000 metric ton botanical raw drugs are this imported and exported, and annual turnover of this more nearly 20,000 crores. So this is a big industry in herbal industry right now, in the not only in India, in the world. So in this, because of this high demand, these medicinal plants trades are lot of having the so many problems. In among this, there are three major herbal drug problems. Herbal drugs problems. One is lack of validation, and that means evidence-based pharmacology, and lack of toxicological evaluations, and may often contains adulterations. So our research is focused on this third one, may often contain adulterants. In the herbal industries, these two terms are common. That means substitution and adulteration. That means what is substitution and what is adulteration. I will give an example. So substitution means this Bacopa monnieri, that's plant, that trade name is called as a Brahmi. A couple of them know about this product. This product Brahmi is used for the memory enhancer, and this Bacopa monnieri is an authentic plant for preparing up this Brahmi. But Centella asiatica is a different plant, but still this plant also used for the preparation of Brahmi, because these both plants contains a biologically similar activity, and this is affiliated with, this is approved with Ayurvedic Pharmacopoeia of India. And sorry, I don't know why this red color came. Okay. This is, and second thing is this adulteration. This adulteration, that means Saraka Ashoka is an authentic plant, but some of the things, some of the other plants, especially Palialthia longifolia and Troma orientalis, also they will called as Ashoka because of this similar vernacularly, not vernacularly, just locally, these plants also is called as a Ashoka. But these plants completely entirely different from the biological and chemical difference. But still these plants, Palialthia used for an adulteration of Sarakosha. This is called an adulteration. Why this adulteration happens? These are all the main problems in adulteration. Confusion in vernacular names between the indigenous system and local names. That means vernacular names and local names are confusing. And second thing is the similarity in the morphological feature. That means looking similar morphological characters, have a couple of plants having the similar morphology characters. And another one thing is geographically co-occurring species. That means that plants is available and closely related plant species also produce in the growing in the same environment and habitat. That is also one problem. And major problem is 80% of the medicinal plants of the raw drugs are collected from the wild. If you are in the wild collection, we need to know about good knowledge about that plant, particular plant, and you know the exact things about this particular plant, then only you can go and collect. Otherwise, sometimes if you don't have the knowledge about that plant, you make wrong with something else. So this is also main problem in adulteration. So as I mentioned, the common names, as I mentioned earlier, Saraka Ashoka and Palialthia longifolia. Because of this Ashoka common name, these plants are also adulterated. And same thing is another one, an example as a Gokru. That this Gokru contain two, Chota Gokru and Bada Gokru. This Chota Gokru means tribulus sclerosis, and this is used for the different, that means liver and kidney disorder purpose, and muscle building and kidney disorder purpose. But in the Bada Gokru, Pedalium murex, they used for the increasing the testosterone level. That means both are different plants and both having the different activity and also different purposes. But still, these are all adulterated with each other. And another one, this most common thing is an similar looking. If you see these plants, both are look like similar. Once only in the flowering time, only in the flowering time only can able to distinguish between the things. If you dry this, both samples are look like similar. This is also one of the major problem in herbal industries. This is one of the classic example happened in Belgium. Because of this common vernacular names, these people are misidentified the plant and used it used instead of that plant. So Stepania tetandra is an authentic plant used for anti-inflammatory purpose, but people are confused with another plant that called as Aristolochia fanchi. This plant contains the aristolochic acid. This aristolochic acid is a mutagenic, carcinogenic, and nephrotoxic. So in this case, these people misidentified with a common fancy name. So more than 100 women are suffering from kidney failure with later stage development cancer for the urinary system. So this study is covered in the Nature. After this paper, so many research talks are going in the herbal industry for adulteration or substitution or else admixture. So how this study is going to address that means what are the methods to identify this adulteration. So generally how we are going to identify this adulteration in the trade. So for visual infection by that means by seeing the and organoleptic examination, that means microscopy and macroscopic examination. This is a preliminary examination. We can differentiate between the plants and herbal drugs. If you see like this, you can easily differentiate among these herbal drugs. But if they're in the dried form, of course we can it will be difficult in the preliminary visualization, but when we're using microscopy and macroscopy, we can easily differentiate. But in case of powder form, it will be difficult. If we need it, very difficult to identify using the microscopy and macroscopy. Of course we can identify still, but if we need for identification, we need expert taxonomist and well-known subject expert, then only we can identify that product. So apart from this microscopy and macroscopy, what are the available techniques? So chemical examination, of course, we can identify the chemical examination using a different advanced chemical methods. But these chemical methods also having some limitation. See this one, this CPT means camptothecin. This camptothecin is producing across all the Icacinaceae family. This C123 number indicates the different species. See, all the species also contain the CPT. So in this case, if the standard marker, if you take as camptothecin, very difficult to identify or differentiate the among the species because all species contain the same compounds. So what are the next other techniques? So next other techniques are some DNA based molecular techniques, that means DNA based techniques. So our research is focused on DNA based techniques only. So we used for earlier, initially we started to work on a DNA barcodes. These DNA barcodes are like unique, like other system, that means unique identity system. So what do you mean by DNA barcoding? The DNA barcoding is a short genetic markers used for the identification of the species using the chloroplast or nuclear genome for the plants and animals, mitochondrial system for the animals. This is an now actually emerging techniques to identify the molecular identification of the plants. So now number of universal barcode regions are available. So different plants, different family wise, different organisms wise, like we have a lot of that means more than 68 to 10 universal barcode primers are there. So in this among this one, we in our studies we selected four barcode regions: ITS, MATK, and RBCL PSBS regions. So this methodology is very simple. This once we have to collect it, once we collect the medicinal plants, authentic medicinal plants as well as the raw drugs. And after the collections, we can sample preparation. That means we can remove the tissue for dead tissues are like this. Then after the tissue, selecting the tissue sample, do for the DNA isolations using the conventional RS commercial available kits. And later we can use for PCR amplification based upon the shortlisting of the universal markers. And later sequencing. After sequencing, we can edit manually. We can edit the sequencing using the bioinformative. Now various tools are available. After editing the sequences, then you can compare with those sequences with your reference library and also existing NCBA available databases. And also we can construct the phylogenetic tree if they're related species also we can identify. So these are just general methodology. So as I mentioned, collection of the medicinal plants is very important in the barcoding, generating the reference barcode library. So once if we collected the authentic medicinal plants, so we need to develop the herbariums. So herbariums means we have to collect at least at least 3 to 5 different geographical locations of the same plant, and we can prepare the herbariums and authenticated with the 2 to 3 taxonomy, the expert taxonomist, and submitted to data any depositories like BSI Botanical Survey of India or else your institutional depositories. That is the most and important thing in the generating the barcodes. After the generating the barcodes, we actually went to the South India, including North India. We went to visiting the marketplaces and we collected a lot of medicinal herbal drugs in the different names and different things we collected. So I will give you an example of couple of our research work what we done. So we selected then the Bala and Mahabala Ayurvedically is most important and also widely available, available and widely distributed these drugs, Bala and Mahabala. That means Sida, according to Ayurvedic pharmacopoeia, Sida cardifolia is a Bala and Sida rambifolia is a Mahabala. Sida cardata is a Nagabala and Butalan indicum is Hatibala. And also related species also we collected as we collected like this. And after the collections we also collected the market South Indian market samples with respect to Bala, Mahabala and Atibala we collected. So this I will go to quick result and summarize the result. We in this work we shortlisted 3 barcode region ITAS, RBCL, MATK, and also we calculated intra-specific distance and intra-specific variations, and we found very interesting result. Once we developed the barcode, our own reference sequences and also available NCBA available depositories, we construct the phylogenetic tree and look at this all the whatever we collected the market samples all are clustered with Sida acuta. That means they whatever they market they're selling as a Sida acuta, though actually those samples are supposed to cluster with the Sida cardifolia, but they are clustered with Sida acuta. So they using this using this PSP TNA markers we found this result. And also similarly we did other regions also ITS region, we found the similar result. So that means around 80% of the whatever we collected market samples are adulterated with Sida acuta. So you can go through the details about this our publication. We can find detailed things. And second thing also we worked on the Saraka Ashoka. This Saraka Ashoka is a very important medicinal plants in Indian context, and this plant is widely used for the gynecological problems. And because of this over exploitation of this plant, now this plant become locally endangered. So another one problem of this plant is only available in the forest lands. This plant is not cultivated, so we have demand is more but the production is less. So that's the reason this plants also now is endangered. We wanted to know the what is the available market, whether they will selling as a current authentic Saraka Ashoka or different. So in this case, we collected the different authentic samples from the various part of the country, and we developed the RBCL and PSBT RNH markers, and we deposited in the GenBank accession. And meanwhile, we collected from the raw drug from the across the India, including the North India, Northeast regions also, around 25 herbal outlets we collected throughout India. So we done the study, and we in this study we selected RBCL and PSP regions. We calculated the intra within a variation among this within intraspecific variation we can calculate it. And this result also found interestingly say See, look at this. This HIS means these are our herbal outlets, and this Sarakasha BRM means our authentic samples. See, 1, 2, and 3 samples only clustered with Saraka Ashoka. Remaining all samples are not clustered with Ashoka. That means only 3 samples are is authentic Ashoka. Remaining all are maybe related, not authentic. It means not closely related species of Saraka Ashoka. So we in this study we worked on two methods in the genomic approach also and metabolomic approach also. Genomic approach we used for two regions, and also metabolic approach also we did for NMR of metabolites analysis, and this see red color indicates authentic plants, and these are all herbal outlets. So in the herbal outlets, all are similarly as showed in the genomic approach. Metabolic approach also found same similar result. Around 80% of the herbal trade, whatever we collected samples are adulterated. That means they're not at all Saraka Ashoka. So this is both in metabolomics and genomic results shows the similar result. So this one also we can find out the complete detail about this in this our publication. And not only this one, these up to here we worked on single ingredients. Then what about if the ingredient contains purple product contains more than one plant species? For example, if you see this one, these tablets contain more than 5 to 6 herbal ingredients, and powder contains more than 10 ingredients. So we use it for that means polyherbal products. 39 samples we used for. This is a recent work using this genomic approach we used for next generation sequence to analyze these samples. So we found again interestingly for around 76% of the samples are found authentic whatever they mentioned in the labels, but remaining 25 to 30% of the samples we didn't able to analyze, didn't able to identify, maybe several reasons including the degradation of DNA and processing of the products and several other factors. But 76% of the samples were only authenticated. So this is also we recently published in the scientific report. This is an, and apart from after all these things, we generated the digital database. That means if you see the Saraka Ashoka, if you put including the trade names and markers, what are the markers and also what are the possible adulterant, all the information we included in the database, and including the images and raw drug images and like this, and macroscopy and other things, and also related sequence and genes data, and we included. So we developed the database and including our own herbariums and also our own raw data and also DNA barcode sequences. We developed the database, and finally we this two regulatory framework we proposed for this herbal drug authentication system. One is national policy for quality assurance for herbal trade, and second one is nationally accredited laboratory to for authenticate and certified raw herbal drugs. So in these two policies, we includes the collectors to consumer. That means collectors and including the manufacturing companies and consumers. That our preliminary intention is to herbal safety. So this process this collection of raw herbal drugs and curation. That means voucher specific preparations and authentication, not only genetic work, including the chemistry metabolites work, whichever suitable methods we can identify and certified with these regulatory agencies. And after certificate, then digitalize this one. That means anybody can access those data and easily can come to know which is the more adulterated or else authentic plants, and including the chemical and genetic markers. So this is also we covered in the herbal drug safety journals. We can find it. And in summary, in our study confirmed that several medicinal plants or groups are widely adulterated in herbal drugs. An herbal drug authentication system has developed that integrating one platform information at medicinal plants, and this HAS could play an important role in the regulating and offering the quality assurance of the herbal trade in India. So, and also our works in especially Saraka Ashoka and other couple of work covered in the media, press and media coverages, including the Department of Biotechnology and many other several national and international news media. So our studies covered in a lot of attention in media for adulteration studies. And also I would like to acknowledge my past and present supervisors and also collaborator. This is an multi-institutional and that means India, Canada and Thailand collaboration projects. So, and also we received a lot of grants from the different various funding agency, Department of Biotechnology and also this Thailand government and also Canadian government, we received the funding support. And thank you. If you have any questions, I can open to take.
Big thank you, Dr. Santosh. And it is a long time I have seen you again. Because time constraint, we already got delayed. Thank you very much once again. And if any question comes, I will post you to the mail. Please, once again, I express my heartfelt thanks. And now I request our Dr. M. Aruna Madam to introduce our next speaker. That is Dr. Insaf Ahmed Qureshi sir from University of Hyderabad. Over to you, Aruna ma'am.
Good afternoon, sir. Am I audible, sir?
Yes, ma'am, you're audible.
Good afternoon, sir. Dr. Insaf Ahmed Qureshi, sir. And we are very much happy to have you today on the platform. And it's my pleasure to introduce you, sir. Dr. Insaf Ahmed Qureshi, sir, who is serving as Senior Assistant Professor at Department of Biotechnology and Bioinformatics from School of Life Sciences and University of Hyderabad. And he has obtained his master's degree from Jivaji University, Gwalior in the year 1999 with applied microbiology as his subject. And he has obtained his doctoral degree, PhD, from Ahmedabad University, New Delhi in the year 2005 in biotechnology. And the thesis of his, the title of his thesis is Isolation and Characterization of Lectins and the Genes from Chickpea, that is Caesar Editinum. And coming to his research experience, he has gained more than 10 years of teaching experience and 15 years of research experience. And he worked as visiting scholar in Department of Biochemistry, University of Missouri, Columbia, USA, from December 2013 to December 2014 and December 2018 to March 2019. And he worked as a postdoc research fellow in Division of Structural and Computational Biology, School of Biological Sciences, Singapore, from February 2007 to December 2010. And he's also worked as project associate in Structural Biology Unit, National Institute of Immunology, New Delhi, from 2005 to February 2007. And he's conferred with many honors and awards. Few of the important ones are, he's selected for esteemed ICMR International Fellowship for Young Biomedical Scientists during 2018 to 19 by ICMR New Delhi. And he's also been awarded with prestigious ICMR Prize 2010 by Indian Council of Medical Research, New Delhi for the year 2010. And he's awarded with coveted Indo-US Raman Fellowship by University Grants Commission, New Delhi for the year 2013 to 14. And he has been selected for esteemed International Exchange Program by Indian National Science Academy, New Delhi for the year 2013 and 14 and 18 and 19. And he has been awarded with Fast Track Project and Young Scientist by Science and Engineering Research Board, New Delhi in the year 2012. And coming to his research publications, he has published more than 60 papers in reputed international and national journals, and he had delivered many invited talks at various symposia and conferences, and he has done and he is also to online projects, and he is with two of the ongoing projects, and he has completed many projects which have been funded by CSIR and University Grants Commission, UGC, and also by University of Hyderabad, and also by Science and Engineering Research Board. So this is all about his research experience, and he's also actively involved into many associations where he is life member of National Academy of Sciences, and life member of Indian Crystallographic Association, and life member of Indian Biophysical Society, and life member of Society for Plant Biochemistry and Biotechnology. And we are very happy to learn that he is Associate Fellow of Telangana Academy of Sciences and also Associate Fellow of AP Academy of Sciences. So with this brief introduction, I welcome and now I will invite Dr. Insaf Ahmed Qureshi sir to deliver his talk on topic development of novel multi-epitope vaccine against SARS-CoV using immunoinformatics approaches. Thank you, sir. And now over to you, sir. Thank you.
Thanks, madam. I hope I am audible.
Yes, sir.
Yeah, I am highly thankful to firstly, madam, actually, who has given this much kind introduction. Actually, I don't think all these things are required, actually. And
Thank you, sir.
To Dr. Abdul, actually, because he was working on this symposium from the last 1 year, actually, and I was, because since I was interacting with him regarding this, so I am really, I can see his efforts which he has spent for this. Okay, so without wasting further time, actually, because already we are running sort of the schedule, I would like to start my work, actually. So today about our recent research, actually, which is on the development of novel multi-epitope vaccine against the SARS coronavirus 2 using immunotherapeutic approach. So before going about our work, I just want to mention a little bit about the protein, actually. So proteins are fundamental components of performing a variety of biological tasks. They are basically built by the 20 amino acids, and we all are aware about those things. So basically this protein is having a particular three-dimensional structure, and that helps to determine its function also. If you go in the hierarchy of the protein structure, so basically there is a 4 steps. One is the primary in a peptide chain. That is a primary structure. And when we talk about this alpha helix and beta sheets, that forms the secondary structure. And when alpha helix and beta sheets pack together and forms a structure that called tertiary structure. When more than one polypeptide chain come together and they forms the quaternary structure. So if you want to look for the three-dimensional structure of a protein molecule, there is two type of the methods. One is the experimental and another is the computational methods. So experimental, like generally X-ray crystallography, NMR and cryo-EM, and computational method like a homology modeling, threading and ab initio methods. So my lab basically is focused on the structural biology of the protein. So what we do, actually, we determine the three-dimensional structure of the protein, and we use those structures in the different aspects, like a drug discovery. Like we determine a structure of protein and we take the inhibitors and we determine their structure in the presence of the inhibitor to see how those inhibitors are binding and how they are affecting the affecting the protein. And generally we work on the human parasites protein, and my lab is basically focused on the Leishmania is a causative agent of the Kala Azar, and that is very dangerous disease, and it is almost killing every year 50,000 people per year, actually. And it is very common in the Indian region like a Bihar and West Bengal. Another aspect which we work, actually, where we apply the protein structures is the catalytic mechanism to understand the how enzymes work. To understand this mechanism, catalytic mechanism, we use the protein structures, and we have determined the structure of ribokinase, pyridoxal kinase, several proteins using this approach, and then we have given the catalytic mechanism. And recently we have started one more aspect, actually, using the protein structure, that is the vaccine designing, sorry. So in the vaccine designing, today I am going to talk about more about the vaccine designing, not these two aspects. Okay, because this is our recent work, actually, and this is maybe good for the common audience, actually. So as we all are aware about the COVID-19, it is a pandemic and going all around, and that is the reason we all are sitting at home and we are not able to meet face to face and we are interacting through the Zoom or Google Meet. So that is the COVID-19, and COVID-19 causative agent is the SARS coronavirus 2, which is a enveloped virus with positive sense and single-stranded RNA genome. This RNA genome encodes for the several non-structural and structural proteins. Because structural proteins, there is basically 4 structural proteins. One is the spike protein, second is nucleocapsid, third is membrane protein and envelope protein. These proteins play a very critical role in the pathogen attachment and pathogenicity of the virus. If you recollect, actually in the 2002 and 2003, we had a similar kind of problem when we were having the another infection that called the SARS, severe acute respiratory syndrome. So if you see the causative agent of the SARS causative agent was also a SARS coronavirus. So SARS coronavirus and SARS coronavirus 2 both belongs from the coronaviridae virus family. And if you see, they are having almost 80% homology. And their mechanism of infection is also identical. Like both goes to the both cause the similar kind of symptoms like fever, cough, cold, sore throat. Because the mechanism of their action is also same, because they go inside the human body and there they goes and interact with the ACE2 receptor, which is present on the human cell. And through this ACE2 receptor, they goes inside the cell and then they start multiplying. So in the case of the SARS coronavirus, people have generated the neutralizing antibody, and that neutralizing antibody helped to stop the endemic and growth of the SARS coronavirus. So what people thought, when this is having 80% homology, why this neutralizing antibody cannot work on the SARS coronavirus 2? So in the beginning of the 2020, people have tested those neutralizing antibody on the SARS coronavirus 2. But unfortunately, they didn't get success. Then they thought, what is the reason why we didn't get the success? So if you go back and see, basically this, because SARS coronavirus, both 1 or 2, both interacts with the ACE2 receptor. And ACE2 receptor, when they interact, there is one particular protein on the SARS coronavirus, that is a spike protein. So spike protein interacts with the ACE2. So what they did, actually, they have generated the anti-ACE2 antibody and then they have blocked this one. But in this case, SARS coronavirus 2, they didn't get success. So then they have checked and they found spike protein is made by the 2 subunits. One is the S1 and another is the S2 subunit. So S1 subunit contains the receptor binding motif. The receptor binding motif play a role with the binding of the SARS corona with the ACE2 receptor. So if you see, basically RBM, receptor binding domain motif, is present in the RBD of the S1 protein. So if you see the S1 protein is having a RBD domain which contains around the 250 amino acids. And that is having a one signature that called the RBM, that is around 75 amino acids long. So if you compare the RBD domain of SARS coronavirus 1 and SARS coronavirus 2, they are having almost 75% identity. But if you see at the region of the RBM, which is going and binding with the ACE2 receptor, that is having only the 50% homology. And this way they realize, yes, this RBM, the difference in the RBM is making this neutralizing antibody not to interact with the SARS coronavirus 2. So on this basis, on this hypothesis, we have designed a methodology. In that methodology, what we did, actually, firstly, we have taken the RBD domain sequence, which is around 250 amino acids, and we have retrieved the data from the PDB database, and we have identified the B and T cell epitopes of the RBD domain. And after identifying them, we have constructed a vaccine. And after constructing a vaccine, we have gone to analyze the different properties of the vaccine, like antigenicity, allergenicity, and physiochemical properties. Then we have determined the 3-dimensional structure of the vaccine candidate. And as vaccine candidates interact with the, when they goes inside the body of the human, they have done the docking study with the TLR3. And then we have performed the MD simulation and in silico cloning. So this one, I will just explain you how we have done actually. So firstly, what we have done, we have identified the CTL epitope. CTL basically cytotoxic T lymphocytes epitope, and they are present on the, they goes and interact with the different receptors in the human being. So what we did, we have taken 250 amino acids and we have analyzed the, we have screened the sequences, and then we found there is almost 4 epitopes which is having very promising result. And among them 4 epitopes we checked their antigenicity, their non-allergenicity, non-toxicity, and the number of HLA alleles which they will go and bind in the human cells. So on this basis, among the 4, we have selected one epitope as a CTL epitope. Similarly, another T cell epitope is the HTL epitope. So we have identified 4 epitopes on the basis of their antigenicity, non-allergenicity, and their allele binding, and the IFN gamma, which is important for the activation of the immune system. So on this basis, we have identified around 4 amino acids. And if you see, and this is a COVID-19 is a pandemic, and it affects the population worldwide. It is not like only in the India or in a particular continent. It is affecting population worldwide. So we have analyzed this population coverage for these epitopes, and we found this is almost covering the 92.5% of the population globally. And it is the most population coverage we have observed for the North American countries, where it is showing the 99.77%. If you see in the sense of the India, it is showing almost the 95% coverage for Indian population. And along, if you see that China is the originating country for the coronavirus. So if you see there, it is having almost 80% coverage for the Chinese population. Similarly, we have identified the linear B cell epitopes, and we found that there is one epitope that is the antigenic, highly antigenic, non-allergenic, non-toxic, and present on the transmembrane topology. Because transmembrane topology is important, if they are inside the transmembrane region, then they will not be exposed for the human cells. And if they will not expose for the human cells, human immune cells, then human immune cells will not able to generate the immune response. So we found among the several one epitope which was present the outside of the surface. Then further we have analyzed this epitope, because other important characteristics are like surface accessibility, flexibility, hydrophilicity, and the beta time prediction. So we found that the selected epitopes, this LQ, SY, GF, QPT, and GV, GY, QP, that is around, that is having 16 amino acids, is showing very good score for the surface accessibility, flexibility, hydrophilicity, and the beta turn prediction. On this, on the basis of all these parameters, we have selected altogether 6 epitopes. So these 6 epitopes, among these 6 epitopes, one epitope is LVL, one epitope is CTL, and another epitope is the another 4 epitope belongs from the HTL. So we have linked these all epitopes using the linker. Linker is required actually because they contribute towards the maximal flexibility of the amino acid to fold into the appropriate conformation. So if you see these epitopes are linked by the different epitopes, different linkers. Beside the different epitopes, we have used HBD2, human beta defensin 2, as a adjuvant, because this human beta defensin 2 upregulates at the time of the lung infection and increase the innate and adaptive immunity. As we know, because the coronavirus main target is the lungs. So this is the HBD2, which is the adjuvant that upregulate during the lung infection. So this one we have used, and all together we have taken 6 amino acids to construct a novel vaccine candidate against the coronavirus. After constructing, we have checked the physiochemical properties of this vaccine protein, and we found this protein is highly soluble, and it contains the all secondary structure, while mostly secondary structures are the alpha helices. After that, we have determined the three-dimensional structure of the protein using ITSR server, and we have further refined this this structure, this model, by the Galaxy Refine tool. And if you see, this structure is highly compact, and it is also having the correlation with the secondary structure. The tertiary structures are also having correlation with the secondary structure, where you are able to see more alpha helices than the beta sheets. Any structure is not complete until you validate that structure. To further validate the structure, we have analyzed through, we have checked the Ramachandran plot of this structure, and we have checked the ROSA server. If you see the Ramachandran plot, only 99% amino acids were in the allowed region, except one residue that was the valine, and that belongs from the your adjoint, not with the epitope. So it suggests the structure is completely perfect, and it can be used for the further analysis. After determining the structure, we have analyzed the conformational B cell epitope, because conformational B cell epitopes will be present on the cells. So conformational B cell epitope is the cluster of the amino acids that are discontinuous in the protein sequence, but present proximally in the three-dimensional structure of the vaccine protein. So we found almost 60% amino acids are 60% amino acids are the part of the conformational B cell epitope. After determining the conformational epitope, as because they have to generate the immune response, so we have checked the binding of this vaccine with the toll-like receptors. Toll-like receptors are integral membrane proteins which express on the guard cells of the innate immunity and generate the antiviral response. So we have tested the three TLR2, TLR, toll-like receptor, TLR2, TLR3, and TLR4. We have performed the molecular docking using a CLESPRO server, and we found among the three, all three shows interaction, but TLR3 is having more binding and more interaction with the vaccine candidate. On this basis, we have picked up the TLR3 and vaccine complex, and we have further analyzed using a PyMol, and we found this vaccine construct is nicely sitting inside the binding pocket of the TLR3, which is a horse-shaped structure. To check the structural stability of this complex, which we found through the docking study, we have performed the molecular dynamic simulation for the 100 nanoseconds, and we found and we have analyzed the several parameters like RMSD, root mean square deviation, root mean square fluctuation, and root mean and radius of gyration. If you see, root mean RMSD and RG shows the more stability and more compactness. If you see the RMSF case, there is a less fluctuation in the side chain when you are having a complex. When any protein, when two proteins interact with each other, they form some hydrogen bonds. To see whether this protein, this complex is having more hydrogen bonds or less hydrogen bond during simulation, we have analyzed the hydrogen bonds. And if you see, the number of the hydrogen bonds has increased within the time of the simulation. When we have checked at the polar contacts level, we found initial structure is having the lesser polar contacts, and the stable complex is having the more polar contacts, and that makes our complex more stable and more rigorous and suitable for the further study. When we have analyzed the interaction for the vaccine receptor complex before the simulation and after the simulation, we found it is having more interaction before the after the simulation. If you see here, it is having almost 36 interaction, which has changed to the 53 after after simulation. And if you see, the most contributing agent is the hydrogen bond. In the beginning, there was only 15 hydrogen bonds. At the end, we are able to see the 25 hydrogen bonds. After confirming after confirming the stability of the complex, we have performed the uh, in silico cloning into the PET vector. PET28 vector is an expression vector for the E. coli. So E. coli is a well-established expression system to generate the high level of the protein, high expression of high level of the protein. So we have used a PET28 vector, and we have cloned this our vaccine candidate, which is around 146 amino acids and around 500 nucleotides are there. So we have cloned this one using ND1 and XHO1 site. And after this, we have confirmed virtually this one, and we found, yes, we are getting the size which is the calculated size. So then we have then we have concluded this study with this remarks that we have developed a multi-epitope vaccine candidate against the SARS-CoV-2 from its RBD domain, and it includes the RBM regions also, using the immunoinformatics approach. These epitopes contain several residues which are significant for binding of viral RBD domain to human ACE2 receptor. The vaccine candidate was highly antigenic, it was stable, and it was soluble along with the non-allergenic for the host cells. The binding study of vaccine candidate with the TLR3 immune receptor establishes the stability of the complex. In silico cloning confirms its efficiency and stable expression into the bacterial system. And now we are in the process to do the in vivo study for the same vaccine candidate, which will establish the potency of this vaccine against the COVID-19. And this study has recently published in the Journal of Biomolecular Structure and Dynamics with the title of Multi-Epitope Vaccine Against SARS-CoV-2 Applying Immunoinformatics and Molecular Dynamics Simulation Approaches. Taking a hint from this work, actually, we have explored the another protein. As I mentioned in the beginning, there is a four structural protein in the coronavirus. One is the S protein, which we have explored to design the vaccine candidate, and there is another protein that called the nucleocapsid. So nucleocapsid protein also we have explored, and we have designed the another vaccine candidate using the nucleocapsid, using the similar approach, immunotherapeutics approach, immunoinformatics approach, and this work has been also recently published in the International Journal of Peptide Research and Therapeutics. And this vaccine is also at the level of the in vivo validation, and hopefully we will get some success, and that can serve for the country and serve for the human society. And with this, I would like to acknowledge my PhD scholars who has worked on these proteins rigorously in this corona lockdown period, actually. And I would like to mention here especially one student who is from the Telangana University, and he has recently completed his PhD, and he's looking for the postdoc abroad. And I am also thankful to my sponsoring agencies like SERV, CSIR, ICMR, UGC, and University of Hyderabad. With this, I am thankful for your kind attention and giving your precious time. Thank you.
Thank you, sir. Thank you so much for your presentation, sir. Thank you. In future also, we will expect a kind favor from your side, sir. Big thank you for the beautiful presentation of current times, what we are facing. Thank you very so much, sir. Now I would like to request Dr. Jalandhar to introduce our next speaker, Mr. Biju Dharampalan, who is CSIR and NICAIR teammate. So Dr. Jalandhar, over to you. Dr. Jalandhar. Dr. Jalandhar.
Hello, sir. Hello, sir. Good afternoon, sir.
Yes, you can introduce.
It's my immense pleasure to introduce Mr. Biju Dharmapalan, sir. Mr. Biju Dharmapalan is a well-known science communicator and educator from Kerala who regularly writes for
Biju,
Science
you can
Reporter.
video.
Sir.
Biju.
Hello.
Yeah, you continue your introduction.
Who regularly writes for Science Reporter and Science India and is involved in various science popularization programs of government. He has also contributed articles to national newspapers like The Hindu, The Telegraph, The Statesman, The Daily Guardian, and also in agriculture magazine like Karshakar. He has more than 10 years of experience in extension activities, science communication activities. In the last 10 years, he has coordinated 3 international and nearly 50 national seminars supported by various international and national agencies like EMBO, DST, KSCSTE, etc. Has been actively associated with the science programs of Radio MacFast 90.4, the community FM radio, and Patamamitta district of Kerala. The weekly bilingual science program, Science Scope, is well received by the listeners. He is also associated with the science talk programs of All India Radio, New Delhi. He was the former head School of Biosciences, MSc He is having nearly 20 years of experience in teaching, research, and is the author of several textbooks and articles. Some books of some of his books are followed as reference text by various universities. He had worked at Malvius College, Trivandrum, TPGRI Fellowed, and CPCRI Kasaragod also. Currently, he is doing his doctoral research in science communication at CSIR, NIS CAIR, New Delhi. An academically meritorious person, Biju Dharmapalan is a university rank holder and a recipient of University Merit Scholarship throughout his studies and has qualified national level exams like GATE and NET. He has also received a fellowship from Indian Academy of Science, Bangalore. He has handled numerous research projects funded by KS, CSTE, Government of Kerala, and National Medicinal Plant Board, Government of India. He has won prize for the state level essay writing competition conducted by Kerala State Council for Science, Technology and Environment. His name is listed in the database of science communicators in India. Published by Vignan Prasar DST, Government of India, and also as a mentor resource person for seminar, winter camps, and other inspired initiatives of DST, Government of India. He attended the first science conclave with the Nobel laureates organized by IIT Allahabad, and also in the round table meet on science communication organized during IISF 2017. As an invited delegate, luminous teacher, he had been invited twice to attend the Indian Academy of Science mid-year and annual meeting as an invited teacher fellow. He was nominated by DBT, Government of India, as a delegate for the National Science Teachers Congress held as part of IISF 2018 at Lucknow. Recently, his name has been nominated for the prestigious P.T. Bhaskara Panikar Science Writing Fellowship instituted by the Kerala State Council for Science, Technology and Environment, Government of Kerala. Out of line, he has published near about 25 national paper articles in national and international journals, and 71 articles in different magazines, and 9 articles in different newspapers, and 14 book chapters in different books, and he wrote near about 23 papers for the conference proceedings, and he has conducted near about 23 radio programs, and he has already invited for the near about 26 different talks. Thank you, sir. Thank you very much for giving me this opportunity. Over to you, sir.
Thank you, sir. Yeah, can you hear me?
Yes,
can
sir.
you hear me?
Yes,
Okay.
sir.
Yes, sir.
Okay. Okay, I am visible also, right? Hello.
Sir. Continue, sir.
Okay, you can see me? Hello.
No, sir. No, sir. No, sir. Your screen is visible, sir. Yes, sir. Yes,
Okay.
sir. Sir.
Okay, sorry. Screen is visible? Hello, sir. Is it visible?
Yes, sir. Yes, sir.
So I think there is some issue when I share the screen. Screen is visible right now, right?
Yes, sir.
Okay, okay, fine. So thank you. First of all, I would like to thank my friend Dr. Halil Khan, who invited me for this prestigious webinar, international conference. So, as you have listened to a couple of good lectures based on science, I'm not going to give an overview of hardcore science, but I will give a superficial view targeting the student community. So, today what I will like to speak is molecular biology for understanding the complexities of life. So as you know, throughout the history, in the geological history, we have different time periods, and we have seen evolution of different organisms in the different time intervals, and in nature you can see different types of diversity of organisms. From mushrooms you can see, you can have different types of pteridophytes, you can see even in gymnosperms you have the living fossils. Ginkgo biloba is there, taxus, the highly medicinal plants like taxus is there. So then when you might have happened to see about this Artocarpus or the jackfruit seedlings. When you grow seedlings, you see sometimes some variations like this may occur in floriculture. You can see different colors of roses. Even blue rose we have able to produce by breeding approaches. You can
You are
see
resounding things.
white-colored peacocks. Hello.
You're getting resound.
Hello.
Your mic is getting resound. You are getting your sound again back.
Okay. How is it, sir? Now?
Okay.
Hello.
Okay.
Okay, fine. So we get okay. Then we have in our chimpanzee. You might have seen chimpanzees. So then in the whole population, if you go through the different facial expressions of the humans, you can see there's a vast number of diversity. You can see in different races. Even within our country, we can see people of. We can have a specific identity of each individual when we go through the phenotype of an organism. I will consider human beings as an organism. So all these things are happening because of one unique structure which is present inside our cell, which we call as the chromosome. So most of you, I presume, are MSc, BSc students and teachers, and you are aware of the history of the chromosome and DNA structure, everything. In this aspect, I would like to quote Carrie Mullis, who is the inventor of a PCR machine. Science consistently produces a new crop of miracles, truth, and dazzling devices every year. So whatever we see or how we approach to study this different diversity in nature is made possible by the tremendous development inventions that happened in the instrumentation sector. So biology progressed not with biology alone. It progressed along with other branches of science and technology, physics, engineering science, electronics. All these branches have made the evolution of our biological system. And we know that this diversity, how to study this diversity. You can have these diversities studied by looking at the phenotype. That is one simple aspect that we have been doing since time immemorial. You can study at the biomolecule level. So as you know, traditionally we focus on carbohydrates, lipids, proteins, nucleic acids. So these biomolecules, we're concentrating on these biomolecules, and among these biomolecules, we mostly concentrate for studying the what we call the history behind the life. We always look at the DNA, which is present in our chromosome. So as you know, the structure of DNA is very well understood, and it is approximately 2 meters in length, and it consists of 30,000, approximately 25,000 genes, which codes for the protein. So when you go through the history of DNA, we understand it starts with the see in every scientific history. I think today it's a sad day for Indian science because the eminent Indian scientist Rodam Narasimham, Professor Rodam, he has passed away. He has written exemplary work regarding the history of Indian science. So when you study science, we have to look at the historical aspect also, because then only we will get to know about how relevant that particular discovery is. When we look at the current scenario, it is very easy to understand science because we are in the what we call highly developed state. But in the initial state, from the scratch, how we developed, that also is something students has to look into. So this study has started in Frederick Misher in 1869. So this textbook, about the famous Griffith experiments with rough strain and smooth strain virus in mouse. Then there is Oswald Avery's discovery of what caused this factor that carried the trait of this virulence trait in the mice. So from Avery's work, then Chargaff studied the ratio of the different bases that is present in the DNA, the nucleotide bases. So the Chargaff rule. So then, using the X-ray crystallographic studies that has been carried out by Morris Wilkins and Franklin. So all this long history helped actually Watson and Crick to crack the structure of DNA. So even though we always acknowledge Watson and Crick as the discoverer of DNA structure, there are many persons, especially Morris Wilkins and Rosalind Franklin, who did a lot of work, the fundamental work for the development of DNA models. So when the moment we discovered elucidated the structure, remember this discovery of structure of DNA happened in 1953. But the human beings and all the living systems were there in this planet from thousands of years. But when you go through the history of science, it is a very what we call we are in this particular field is a very young field. We have history starts with 1953. So when this particular DNA discovery was made, Francis Crick in the paper, he has mentioned on aspect that he has spotted. It has not escaped our notice that the specific pairing we have postulated immediately suggest a possible copying mechanism for the genetic material, signifying the importance of semi-conservative replication. So you know about DNA, then we know about the proteins. So even though you know that whatever is happening in the structure of DNA, or especially the chromosome, the functional part of the DNA, when it becomes a chromosome, because of another very important molecule, proteins. So without proteins, I don't think DNA has got any existence. Proteins are the real workhorses in our cell. So protein sequence can also be used for infer the evolutionary relationship. So this whatever is happening inside a living cell, the DNA, the protein, and their interaction. See, all these forms the what we call basis for and proteomic studies. So these students are so, but when you say that there's proteomics and genomics, how you are studying these organisms, how you are studying these mechanisms, sorry, how this mechanism. In molecular biology, the fundamental central dogma we used to say is this that is called from DNA you have the RNA, from RNA you have the protein. So this is the central dogma. But, in today's world, I will not say this dogma is correct, because you can have the reverse process also possible by synthetic means. So, but sticking to the central dogma, we know that in a molecular biology lab, when you these biomolecules, we may have electrophoresis, we may have gel dock system, you will have various types of mass spectroscopic techniques, you will have XRD techniques, you will have SEM, TEM, you will have, this is multi-top. Then a very important aspect, mechanism that we use in molecular biology is polymerase chain reaction. I would say this is one particular that revolutionized the field of biology per se. So this is invented in this got a Nobel Prize also for Candy Mullis. So PCR is a lab cell. So this is so whatever DNA synthesis it happens in a cell is happening in a PCR machine. So this is one of the earlier models of old model of PCR machine, but now you are not getting that type of PCR. So, this particular PCR has been mentioned in the Science magazine as the what you call as a molecule of the year. These are elementary things. I know you know the different steps of a PCR reaction. There is denaturation is there, annealing is there, extension is there. So fundamentally, what happens is you are doing something which is happening inside the DNA synthesis happens inside the cell, as you know, in the S phase of the cell cycle, artificially doing in a machine. So this particular discovery actually it has become a powerful tool in molecular biology. Remember, molecular biology is not an advanced branch of science. It is a fundamental science which is used to solve every problems in science. So when you do any plant systematics, one of the speaker was telling about taxonomy, which many people are forgetting about taxonomy. That is in a way it is correct. But the taxonomy that we used to practice in olden days and today are needed some kind of difference. So the morphological taxonomy alone will not suffice to cater to the needs of today, because in today you know that DNA barcoding is there even in airports. By simply scanning, custom people can take away or find out whether an endemic, extinct, or endangered, sorry, not an extinct, any endangered or threatened plant is being transported across the country. So these things can be identified by the help of one particular instrument is the PCR. It has been widely used in diseases also for diagnostic purpose. We have seen PCR reactions using in we have used it in HIV. Nowadays, we know that in SARS-CoV case, uh, this is the new version of the PCR, RT-PCR. Then in SARS-CoV, in this particular pandemic time, many people have been in media also. We hear a lot of things that is RT-PCR as a diagnostic tool for detecting SARS-CoV among the population. So this particular technique coupled with another technique, which is the DNA sequencing, which also an important technique which has been used to solve biological issues. So as the traditional methods goes, we have a chain termination or dideoxy method put forward by Sanger, and we have Maxim Gilbert sequencing techniques, and we have the next generation sequencing techniques like pyro sequencing. I like thing, but mostly that's when you say sequencing, we have been using the Sanger Colson method, which has been standardized during 1970s, which is dideoxy chain termination method. This method is being used widely, and this is also converted into the automated sequencing technique, which was being used till a few years back. But so this is the output of the DNA sequencing in dideoxy chain termination method. This again is an output. So this Maxim Gilbert method, we get the output in the auto radiogram. So this person is holding. This is from Rajiv Gandhi Center for Biotechnology, Trivandrum. So this is one of the earliest pictures of the auto radiogram of Maxim Gilbert sequencing method. Right now, nobody is using this thing. But these techniques, these sequencing techniques, whether it is Maxim Gilbert or whether it is dideoxy chain termination methods by Sanger's method, all these methods have a limitation, like they are low throughput due to the preparation, and it's what you call, it's a tedious process. Basically, these are all tedious process. So people see when technology evolves, we want everything in a fast. So nobody can wait for 2 or 3 days. The technique, we want the every reactions to be analyzed quickly within nanoseconds if possible. So that is the time frame we give it in science in present day. So we have the next generation sequencing techniques evolved. We had because of lack of time, I'm not going to explain everything in detail. So we have the Illumina sequencer, we have the Roche, we have Solex sequencer, this pyro sequencing. Secondly, you know that in each evolution of these techniques, people are trying to simplify the identification of analysis. It is not complication, because it's simplifying. By when a nucleotide joins, it evolves some kind of energy. So whether you can detect that energy in the visible spectrum. So these are the fundamental questions people ask, and in today's world, many people are using is Illumina. They have all the next generation sequences. Then there is a solid sequencing by ligation steps. then we have semiconductor sequencing, we have the single molecule real-time sequencing. Then this is very important, nanopore sequencing. Uh, this is standard by the Oxford Nanopore. So this is the machine. It's a very simple. It's a you can keep this machine in the pocket, and you can just load your DNA sample here and connect it to your laptop, and you get the sequences within seconds. See, in earlier times, it used to a tedious process of maybe 1 month to sequence an organism. So or DNA. Now, right now, within seconds, you can sequence DNA. Uh, this is the evolution of technology. So this is an outline of various first generation and next generation sequencing techniques. So basically, in the next generation, the sequencing are very fast, and read length is high. So when you compare the sequencing labs, you will understand this is an sequencing lab which worked during the time of Human Genome Project in 1994. So where you had very sophisticated instrumentation, many people are working together. But when it comes to 2000, even with a single person can handle all the sequencing efforts. So based on this sequencing and PCR based techniques, many works are being seen. If you go through the literature, any biological work, these two instrumentation, these two techniques, sequencing and pure polymerase chain reactions, are widely used. Without these two, I don't think in today's world, life science has got any existence. So, so they coupled with PCR and human genome this, sequencing machines. We have this Human Genome Project. As you know, in late 90s, it was a very it was in very it is the Genome Project. So as you know, the history shows that it started in 1985 by Robert Sismier. It is regarding a Department of Atomic Energy US discussion. It's a preliminary discussion, but from there, this particular consortia, Human Genome Project Consortia, was formed in which was initially headed by James Watson, then took over by Francis Collins. Similarly, in different this is in US and in other part of the globe in Britain or UK, we had John Sulston, and also in the US we had a private who is always considered as a maverick scientist, who's a private player, Craig. He has 7 economics honor. So these people together because this is the first time when in science there is a public opinion of what you call a collaboration of and also being making humanity. So there was a difference of opinion by Craig Venter and his counterpart, consortia, but they came together for the sake of humanity. And in 2000, the Human Genome Project was the first draft was announced by Bill Clinton, US President, and British Prime Minister Tony Blair simultaneously through video conferencing. So these techniques have made tremendous changes in the field of diagnostics, and also when you say most of the time we focus it only on human side, but we have this sequencing and PCR in plant sciences also. Many crops have been sequenced, and we have been doing a lot of analysis to study the what you call the evolutionary history of each individual plant to identify a specific trait. when it comes to the India missed the first human genome project. You know that yeah, right now we are in a better position. Most of our labs, even in Hyderabad Central University, they have an excellent facilities. In IGAB, they have excellent facilities. But in 2000, we were in a what they call in the lower side of the science. But still, when it came, there was an initial boost for Indian genomic research came by the publication of this very important paper, Reconstructing Indian Population History by none other than Lalji Singh and Dr. Tangiraj of CCMB Hyderabad. So this is a revolutionary thing in Indian genomics because this is the first collaborative work we had in Indian history. It came in the pages, the cover page Indian Science. But later, there is another scientist, this again a CSIR lab, Sridhar Shivasubbu from IGIB New Delhi. So this is Sridhar, is a young scientist, Sridhar, and his PhD scholar, Dr. Vinod Skaria. So these two young scientists, they are the first from India to sequence an Indian human genome. So from there, we started the Indian genome, and right now we are in that. You know that last year our health minister Harshvardhan, fortunately, I also became a volunteer for that particular work. So now you know that the human genome code has been unraveled, and the concept of even divinity is in a question. Again, who is the creator is always we give creator to the God, but now it's a question mark for the religious thinking. But again, science and religion can it's a different sides of the coin. So this is an outline of this generation sequencing application in crop genetics and breeding. So as I mentioned, for identifying candidate marker genes we are using, and for studying the evolutionary history we are doing it. In every part. So without this so you cannot isolate sequencing and PCR from plant science anymore. In plant metabolic engineering, in the earlier talk, we are listening to secondary metabolites. So these secondary metabolites, you know that you cannot harvest these secondary metabolites directly from the plant. You can, but when you harvest these secondary metabolites from the plant, the problem is you are destroying the plant. So there should be some alternative methods, and plant biologists are trying to produce the same secondary metabolite in plants, in vitro plants, by plant metabolic engineering. And there are a lot of success stories in this field. So in this year, we know that so gene editing is one of the what we call focus area of scientific research, which is widely used in every field of biology. So this year, Madam Kamali Carpenter and Jennifer Donna, they received the Nobel Prize in chemistry for developing this particular method of gene editing. So this gene editing is, see, you know that by the current trend in biology, these more advances in all this molecular biology, as I mentioned, will have a lot of implications in the human system and human population. There can be the genome editing has shown that we can have designer babies as is possible. See, Human Genome Project has visualized the personalized genome medicine. So we had, we will say 60% or 70%, we achieved that goal in human personalized, even though we cannot say it as a personalized medicine, but most of the medicines that is coming into the market is based on some data from the genomic approach, because the medicine which is suitable to the Indian population and to the Caucasian population, different races, has shown slight variation. So these things even pharmaceutical companies are taking into consideration. So the designer babies, it is possible. It has already happened. As you know, the Chinese scientist Jian Qi, he has produced the first gene-edited human babies. But unfortunately, as per the what you call the current rules and regulations, it is not possible to do this kind of work, and he has been sent to jail. But he, as per the reports, he has created the first human gene-edited human beings. So in science, what we find is many things which were not acceptable today will gain acceptance tomorrow. So I will not say that what this scientist has created, even though in today's rules and regulation he may be not acceptable, but remember in future this may be a common thing, because you know that gene editing is widely used to develop pets, characters in plants. So then why not human beings if there's a chance of getting any genetic diseases? So these are all some ethical questions. Ethics again, in biology, my acceptance or my accepting a view may not be acceptable to you. So it is again, it's not an fixed thing. It's ethics. It's change from person to person, and of course, I will say that in future also, time will also change the rules and regulations of ethics. See, we are as a human being. We know that we have conquered this particular planet, and we know in order to save our species, scientists are madly looking for an extra planetary space to start the human colonies. So now you just think about what will happen when we have a space colony, which I presume will get in a realistic mode by another 30 to 50 years. I think most of the youngsters will be able to see that space colonies. So if that happens, because it is a need of the hour, as you know, the human extinction in the near maybe thousands of years after this, but it's a fact. We will become extinct. So in that situation, the scientist wants to save our species. That's why they are building all these space colonies. So in such a case, suppose you want to imagine your sister is in Mars, and you want some medicine, or you want to diagnose a disease, and you want to send a medicine to that your sister in Mars. So how is it possible? Because you cannot travel. Yeah, it will take long time. What scientists are trying to do is they are trying to sequence the DNA in the Mars, and digitally transfer the DNA to that sequence to. So these concepts have been really worked out by this again the old maverick scientist Craig Venter. He has lot of these maverick ideas. He has started a new company, Synthetic Genomics, and uh, uh, he has developed a digital biological converter or biological teleporter. So if these things becomes reality, you can imagine what will be the situation. These things will happen in the near future. So these things have become possible only because of, I will say, this evolution of PCR and DNA uh, synthesizer is also there, and also sequencing efforts. So with these things, I would stop the presentation. Having this particular field of molecular biology is ever evolving, and new techniques are being incorporated, and we need the support of molecular biology in every field of life science. So this will again bring prosperity to not only to humanity but to the whole biological system on this earth today, and in the other space, in the outer space tomorrow. So with this, I would stop my presentation. If you have any questions, I'm happy to note it. So once again, I thank the organizers for giving me this opportunity.
Thank you, Biju. Thank you, because we are running short of time. Our next speaker is busy in meeting with Collectorate of Davangere. So, Aruna ma'am, I request that Dr. Tippeswamy sir will be back after his meeting with Collectorate, collector of his local. Now, I would request Dr. Harish to start his.
Support of molecular biology.
Dr. Harish?
Sir, will it be today?
Yeah, it is expected that he will be back. He just now in his meeting, collector has arrived, and he was saying that he will be not in a position. Acha, he said
Maybe not today.
Okay, he's back now. He said, okay, he's back now. Said, ma'am, you can start his introduction.
Tippeswamy is?
Yes.
Okay, thank you, sir. Good afternoon. I deem it as a privilege to introduce Dr. M. Tippeswamy, who is serving as Assistant Professor at Davangere University, Karnataka. Seeing to his academic profile, it's noted that he has completed his undergraduate program at Government Science College, Tumkur, and he has obtained his Master's MSc in Botany with Environmental Plant Physiology as specialization. And then he worked as a Senior Research Fellow in the project Characterization of Cashew Germplasm based on photosynthetic efficiency tolerance to drought at CPCRI ICAR, Vittal, Karnataka. And as a PhD student, he worked on molecular cloning and characterization of stress-responsive genes in safflower subjected to water stress. And during his PhD studies, he worked at ICGEB, New Delhi, that is International Center for Genetic Engineering and Biotechnology, and is also involved in construction of stress-subtracted seed libraries in various crop plants using bioinformatic tools. And he analyzed the nucleotide sequences and submitted around 2,000 ESTs to NCBI, that is National Center for Biotechnology and Information. As a postdoctoral fellow at Israel, he worked on potato transgenic transcriptome analysis in order to decipher the steroidal glycoalkaloids biosynthetic pathway. And then he joined in the Department of Biochemistry at Indian Institute of Science as a UGC Kotari postdoc fellow and worked on triacyl glycerol biosynthetic pathway in a model plant Arabidopsis. And he's also conferred with awards, few of which are he stood in third position, that is, he got third rank during his master's MSc, and he has got his SRF and SRF from Netherlands, and DBT postdoc fellow, and ARO postdoc fellow from Israel, and he's also recipient of Dr. Kotari PDF award from UGC for 2 times, and has also become a research associate at ICR, IIHR, NICRA. And the research results have been published in national and international reputed journals, and he attended several national and international conferences and seminars where a few of the best presentations were recognized. And he also attended refresher and training courses. And very recently, he was awarded with Summer Research Fellow from Indian Academy of Sciences. And with this brief biodata, I thank Dr. Tippeswamy, who has accepted our invitation. And now I invite Dr. Tippeswamy to deliver his talk on topic RT-PCR and its applications in crop plants. Thank you.
Ma'am, thank you very much for my brief introduction.
Thank you, sir.
I am not able to share my screen. It's showing host disabled attendee screen sharing. So I am troubling in the screen sharing.
Now you can share the screen, sir.
Ahmad, can you resolve this?
Yeah, I have enabled you. Making co-host means you can share the screen actually.
Okay. But it is not taking here. So one photo is coming.
No, but in my screen I have pinned you. Your name is displaying on the screen.
Yes, but I am sharing in my laptop and tracking with my mobile. So two are there.
Wait. I am looking one more. See, you are having three things here. I am making three also co-host. Wait.
Okay.
See, which one is possible, you can, whichever possible you try, because with three things you are joined here.
Yeah, you are subscribed, but it was hang up over here. 1 second. Sir, if don't mind if another speaker is there, let him talk. Next session I will join.
No, your three things are co-host now.
Yeah, but it's what we call our internet is not supporting. It's ended with one a photo.
Achha. Dr. Harish?
Yes, Dr. Ahmed.
Yeah, can you start sharing your screen? I will introduce.
Sure.
I'll start your introduction.
I think
it is my proud privilege. What? Any difficulty you face?
No, it's fine. No problem.
Okay, then you can share your screen and
Yep.
your video. It is my proud privilege to introduce Dr. Harish Handrul. He is a stem cell biologist and tissue engineer inspired by cutting-edge developments of science and technology. He's focused in applying skills to develop 3D printing-based cell and tissue engineering applications for regenerative medicine. His leadership skills demonstrated 6+ years of stem cells and tissue engineering research. Currently, he is working towards building potential in vitro bioengineered tissue organs using 3D bioprinting platforms. He gained experience with leadership qualities to work as team with engineers, doctors, and scientists in contribution towards development of stable platforms of human MSCS, 3D engineered tissue construct from stem cells, and for technology transfer along CGMP quality. Excellent skills in networking for broadening collaborations. He's collaborated with multiple scientists for enhancing the technology and bringing the cutting edge platforms. Skilled in managing multiple tasks, handle multiple projects at time. He was awarded PhD in stem cells and tissue engineering, Faculty of Dentistry, National University of Singapore, and he was awarded Masters in Science in Biotechnology, Kuvempu University, India. His bachelor degree was awarded by Bangalore University. He has got a patents for 4 patents: vascularized tissue, skin, and mucosa equivalent. A rapid method for obtaining high purity embryonic stem cells from mesenchymal stem cells. A method for culture of embryonic stem cells using fibroblasts extracellular matrix derived from embryonic stem cells. Patent application was submitted, and in vitro vascular tissue equivalents from human embryonic stem cells is also patent application submitted. He has got number of publications in the international journals. More than 18 publications he have, and articles under preparation are 5, and he has research experience as postdoctoral fellow in National University of Singapore, visiting postdoctoral fellow at University Health Network Canada, graduate research student in National University of Singapore, and he has grant writing experience for SFS Alternative Protein Seed Challenge Grant, MOE Academic Research Fund Tier 2, NAMIC grant, bioprinted vascularized skin tissue, CRP NRF grant for bioengineered immunocompetent model for drug screening applications, NUHS research grant. He has got professional experience as entrepreneurial lead, lean launchpad in National University of Singapore, vice president research in Hangzhou Wang Sheng Biotechnology Co Limited in Singapore, board member, PhD, CEO, cheeky scientist, and he is scientific advisor for Cellcraft Private Limited. He is technical consultant for Celliviate Technologies. With this introduction, I request Harish, Dr. Harish, to present. Over to you, Dr. Harish.
Thank you, Dr. Ahmed. Thank you for your brief introduction. I would like to thank first with Dr. Ahmed for introducing me and for inviting me for this talk. And also I would like to thank Telangana University for arranging this wonderful conference. So let's start with the talk. So I hope everyone can see and hear my voice, right?
You are very clear.
Okay. So my current topic is on stem cell and additive manufacturing for the next generation of pharmaceutical screening platforms. So as per my introduction, I'm more oriented with the stem cell and tissue engineering background. So just to brief around, I will start with the what is stem cell and how does it work and all those stuff. Okay. So stem cell is specifically like two types of stem cells which are specialized to become any cell type, and also it can have an ability to become by itself by another stem cell. So that's the reason like they call it a stem, because it starts from the stem, and also it can have multiple branches like how the tree's stem has. So that's how the term coined as stem cell. There are three types of stem cells. One is pluripotent stem cells, multipotent stem cells, and specialized stem cells. Pluripotent is basically from embryonic origin, multipotent is basic from adult origin, and specialized is basically the derived of the adult stem cells. The timeline and the key dates which were developed in the development pipeline of the stem cell based therapies is basically the first stem cell isolation was reported in 1998, human stem cell, and major breakthrough happened in 2007 by Amanaka and Thompson, which published the generation of human iPSCs. And recently, like no, though not recently, just before I joined and I introduced to this topic in 2013. So in 2012, Amanaka and Gordon Keller John Gordon got the Nobel Prize for reprogramming stem cells towards the pluripotency of the stem cells. So moving forward, how we can use pluripotent stem cells as a research tool. So since they have the ability to differentiate itself into different type of cells, we can use them as an animal like alternative animal model to have an different type of cells like bone marrow cells, adipocytes, and many other type of cells. Okay. So yeah, so why basically pluripotent stem cells? Why not adult stem cells? So adult stem cells has limited number of passaging or the long life the life of the cells, and also it depends on the origin of which adult tissue they have been isolated. Either it may be bone marrow, or it may be liver tissue, or it may be skin tissue, whatever. So like in terms of pluripotent stem cells, they can be maintained for unlimited number of times, an unlimited number of like passage numbers, and they can convert themselves or reprogram themselves into all the cell types which are available in the human body, like all the 210 cell types, and more standardized differentiation protocols are available to differentiate and pluripotent towards the actual stem cell. So for different type of how we can use these stem cells for disease modeling and therapeutic applications, basically These stem cells, as I said, they are origin from either human embryos or somatic tissues. These can also be transferred by embryonic stem cells and nuclear transfer ESCs, and also induced pluripotent stem cells. Okay, moving forward. What's the issue? Can you see the screen? My screen is not, I don't know what's wrong with this. Okay, so in general, in potential applications of patient specific stem cell, like we can even use patient blood to convert them into patient specific induced pluripotent stem cells. Here is iPSCs, induced pluripotent stem cells, and we can even differentiate towards a specific target cells like cardiomyocytes or even endothelial cells, where we can screen the drugs and customize the medical plan for that specific patient, or even disease modeling can also be done by for the regenerative therapy applications. And this is one part of this is through the differentiation method. Without the differentiation, we can even go for the mutation by gene editing, and also the understanding the sequences, and also use for the specific regenerative therapy for genetically matched healthy cells. So this is the roadmap of the stem cells, iPSC to drug screening. I hope can be visible. So basically, I don't know why the screen is showing like this. Are you able to see clearly? Hello? Hello?
Yes, sir. Go ahead.
Yeah, I don't know. The screen is not okay. Let
It's
me
fine. It's
stop
fine.
and
Go ahead. Go ahead. It's showing fine. It's fine.
It's showing fine.
Yeah, it's
Okay.
fine.
Yeah. Okay. Great. Because for me it is not showing good. That's why. Yeah. Fine. So for the roadmap of the pluripotent stem cells to drug screening, so generally if we choose a specific disease, and we have to choose a specific cell type which we have to be reprogrammed, and then the reprogrammed cells will have a specific factors, transcription factors, where we have to reprogram them for by switching on and on of those factors to have a specific cell type. Either it may be neuron cells, or cardiac cells, or even the skin cells, which can be tested for the healthiness of those cells in an mouse model, and tested the epigenetic analysis. After this, we can even use these cells for the differentiation to relevant cell types, which can be used for the disease phenotyping characterization, and also finally for the drug screening assays. So the second figure is almost the same like what I have said. So we can use the patient derived fibroblast and then reprogram towards the induced pluripotent stem cells, and then differentiate towards neuron, and then go for the patient specific drug candidates, or even for the high throughput screening. Okay, so moving forward, the current clinical status of the pluripotent stem cells is for like there have been clinical trials going on the age-related macular degeneration studies using the iPSCs, and converting them into differentiating them towards the retinal pigmentum epithelium for specifically for the neurogeneric diseases like Parkinson, or spinal cord injuries. IAC and iPSC derived cells have been taken to differentiate, and they are in the clinical phase 1. In fact, in Japan recently, one of an human patient has been introduced with the iPSC derived neural cells for Alzheimer's disease, so that it's in clinical phase 3 stage. And then, so this is just the summary of how the pluripotent stem cell technology can be used in pharmaceutical industries. So basically, for consideration, we have to choose in somatic cell isolation, then iPSC regeneration, iPSC generation, considering the safety, pre-programming, and high quality of iPSC clones, and then considering IPC differentiation for CGMP protocols to have an proper quality IPC derived cells, and then which can be used for the allogenic or autogenic transplantation for drug discovery studies. So apart from this, so along with this, now we are working with a new technology of bioprinting. One second, there is some issue with the yeah. Hello.
Hello, Dr. Harish.
Yeah, wait. There is some issue with the Zoom. Maybe I will stop the share and then I will reconnect it, because I don't know. I am not able to see the screen. Can you see it now?
No.
I don't know, because my screen went blank. And I don't know. Sorry for the interruption. I don't know what's happening with my computer. Still the issue? Like, can see the screen now?
No.
Oh my God.
Dr. Harish is working on his computer. He is having some technical issues. He'll join soon. Please bear with us. Dear participants, since morning, so many people have joined and left the Zoom platform, and I have to make couple of announcements. Like some people have given their email IDs in the registration form, and email is the only source for us to communicate you. Some people are already there in our earlier existing WhatsApp groups, but there are some people who are not there in the WhatsApp group, but still we are communicating them through the emails. In the morning, we have communicated you a mail where Zoom link was shared, and we have even shared the YouTube link. So our team is working on the attendance, but still it will be very difficult to sort out from last two meetings we are struggling like anything about people, because in the registration form people are writing student like that. Yeah, Dr. Harish, you can join. You're back, Dr. Harish. Some people like Rakshita and DUPDANG, Dubding, Lakhavat, and BS Anusha, Deepika, Vasuki, Devendra. These people have given the email ID wrong and their mails are getting bounced. So people who are given wrong email ID, please you check it out and send to us. Dr. Harish, you can start.
Yeah, thanks, Ahmed. Thanks for unmuting me. Sorry, everyone, for the interruption. There was some issue with the computer. Yeah, so we were here for the summary of the technology, pluripotent stem cell technology in pharmaceutical industry. So as I said, like we have to consider about the somatic cell isolation. In that, like we have to consent, we have to take the informed consent from the cell donor, donor privacy, intellectual property, GMP cell manufacturing, and iPSC generation, consideration for safety of reprogramming strategies, and also for the high quality iPSC clones for potential genetic modification, and also as well as the donor privacy and intellectual property. So we have to consider about iPSC differentiation for of standard GMP protocols to be followed for safe iPSC differentiation process, and also for the quality control of the iPSC derived cells. In consideration for the autologous and allogenic and autologous transplantation studies, we have to think for the we have to consider for the GMP standard protocols for transplantation of iPSC derived derivatives, and also informed consent from the cell recipient patients, and also for the auto transplantation and donor recipient are the same for the patient privacy. So along with this, like we can even return scope for the using the iPSCs for basic research, toxicology studies, drug discovery, and also disease modeling and cell therapy applications. So moving forward, now we are developing our technology to make into more mass scale, and also for the large scale production using bioprinting or additive manufacturing technology, where we actually biofabricate the living structures in tissue using tissue engineering concepts, using a set of like like biologically important materials, which can be programmed using the computer aided softwares, which is connected to specialized 3D printers, which helps to biofabricate the tissues based on our desired shape and size. So just to let everyone know that the bioprinting concepts components are majorly of cells, which is bioink, hydrogel, which can be a biopaper, and also the bioprinter, so which helps to develop an bioprinted tissue or organ. So in this process happens in three phases: pre-processing, processing, and post-processing. There are different types of bioprinting methods. So in that, like acellular and cellular bioprinting happens, like cell laden and non-cellular functional scaffolds, where extrusion based bioprinting is most commonly used in cell laden constructs for live tissue development, where it happens based on the pneumatic and mechanical or even electromagnetic driver. So where the cells with the bioink is deposited in a fiber fashion, which actually has the cells inside the bioprinted construct. And also we can use that for the culturing for further cells to mature towards the tissue. So this is how another sort of like more detail how the bioprinting, cell laden bioprinting works. The cells is mixed with a polymer which is cross-linked with a cross-linker, and which forms ultimately an bioink. We can even add the functional biopeptides or growth factors, which enhances the cell to mature inside the bioink to form an towards the tissue. So as I said about the extrusion bioprinting, so it uses the pneumatic pressure, which develops an fiber-like pattern. And in inkjet based bioprinter, it's basically like inkjet printers where they use an dot printing, where in this it uses a piezoelectric actuator or heater, where it drops an bioink in a droplet fashion and in specific programmed design. And this is again, the other one is the laser assisted bioprinting, where they uses the laser, and also this also prints the bioink in a droplet fashion, which could be used for tissue engineering to build to regenerate the tissues. And also those tissues can be used for the drug screening and as well as the in vitro disease modeling. So how is the bioprinting will be having an impact on pharmaceutical industries is basically on testing the efficacies for high throughput screening for isolated targets and in vitro screening model, by replacing the in vivo screening or reducing the use of animals for during the in vivo screening process by using the in vitro screening of bioprinted tissues. And also we can even use the for the high throughput screening of tetragenicity and even metabolite mediated toxicities for using bioprinted models for screening the retrospective screening of the target models. And then moving forward, just for having a wide scope of how the bioprinting scope and opportunities, we can actually bioprint do the organ printing by using the specific tissue cells like liver cells or even the cartilage, bone, skin, and even the cardiac tissues as well. This is this bioprinting for drug discovery and development in pharmaceutics, how we can use it for drug screening, drug delivery, and ADME assays, and even for the high throughput drug screening testing. So this is the future which we bioprinting and tissue engineers believe on this, on having a personalized future medicine. So where an patient goes to a doctor for diagnosis, and doctor will give a digital prescription which is designed which is connected to an server, and server will design an specific sort of design or a pattern which prints like sort of tablets or even medicines or even tissues with a specific concentration of what it is designed for, and that can be personalized, will be applied directly for a patient for an personalized therapeutic outcome. Okay, so this is the just summary of like how the 3D medical application of the 3D bioprinting, either it may be for the bioprinting of tissues or organs, or even for the unique dosage forms, even for the personalized drug dosing and complex drug release profile, and even for the anatomical models of surgical preparation. It is already published, and now it's in national phase. One of an German company is planning to license they are evaluating to license it. So this is one of our major work. And recently we published an paper in Biofabrication Journal of fabrication of vascularized tissues having under chemically defined culture conditions. And we also published last year on our team published last year on the 3D printed PCL PPY scaffolds for regeneration of peripheral nerves. So this is about my work. This is the same where we have, this is the setup for the 3D printing system, and this is the printed scaffolds on which we have grown the neural cells towards the peripheral neurons, which have shown the neural markers expression. So with this, I would like to thank everyone for your attention, and I'm happy to take the questions. Thank you.
Thank you, Dr. Harish, for introducing such a new latest thing, what we don't know, bioprinting. So it was an eye-watching presentation by you. Now we are running short of time. Kindly excuse us. Dr. Tippaswamy sir, are you ready now?
Thank you, Dr. Ahmed.
Thank you. Thank you so much. Sir, are you ready? Sir, you are there in the group, Zoom group. Are you ready, sir? Dr. Harish, excuse. We want to take your snap because we are going to give a press note tomorrow. So
Okay.
you switch on your video. Since long time, my team was keep on asking why Harish sir is not keeping his video on.
Oh, okay, okay.
Dr. Srinivas, please take his snapshot because he was keep on reminding me 3 or 4 times that why Dr. Harish is not visible, why is not visible. So thank you, Dr.
Yeah,
Harish.
sure.
Sir, Tippeswamy sir, which one is yours correct this thing? So I am making co-host for all the 3 things again. Kindly you check it out. Which one can work out? Yeah, now your name has come. Sir, you can start sharing the screen. You're
Yes, sir.
audible.
Now it's
You're audible also.
yes, sir. Yes. Sorry, it's all in because of the network, I think. Now the slide is visible, I think so.
Yeah, you are visible. And you
yes.
your video also on, sir, because our people are
Yes,
there.
sir, yes, sir.
Okay. Why my face is coming? Yeah, okay.
Sir, I think hope everything is there.
Yeah. Thing is visible now.
Start now. Ahmed, thank you very much for the kind opportunity. Explore my knowledge. Introduction, madam. Once again, apologize. Again, I apologize. Start at the right time. Now I will today my PCR and its application in crop plants. So this is my talk. Thanks for the opportunity. So before going to start the RT-PCR and applications, there is a small introduction about. Sir, I am audible.
Sir, you're audible. You're audible.
Yes, sir. Yes, sir. Thank you, sir. Now it will no interruption now onwards. So the world population is expected in the year 2050. It will be reach 9 billion. So for this increasing population, there is a need to cater the food, fiber, fuel, and feedstock needs for the increasing human population. So for this, there is a challenging for the agricultural scientist to create the to feed the growing population food. So sustaining production potential is possible by engineering the crop plants against biotic and abiotic stresses. So these two stresses are the major encounters for the plants to getting the major yield in the food product wise. So the major emphasis is to improve crop productivity, biotic and abiotic environments. So the gene expression analysis is an important tool for identifying key genes and understanding the complex biological process. such as so many metabolic pathways, signal transduction, and plant development. Hello? Hello?
Sir, you are audible, sir.
Yes, sir. Some interruption I heard. That's why, sir. Okay. So, in this direction, the today's topic, that real-time PCR and its applications. The real-time PCR, the qualitative real-time polymerase chain reaction, is a most effective tool. It is a simple, specific, and very sensitive also for quantifying the expression of target genes, especially for the tolerant genes, or drought tolerant, or biotic and abiotic stress tolerant genes. If you want to quantify, there is a proper tool is required to quantify those genes. Various factors such as RNA integrity, reverse transcription efficiency, cDNA quality, primer specificity, amplification efficiency, and selection of even reference genes also may significant influence the reliability of qRT-PCR results. So all these things I will come step by step. What is this qRT-PCR, RT-PCR, and all those things. So the PCR, RT-PCR, and RT-qPCR tools are being used to characterize the differential expression of genes in crop plants. How those genes are differentially expressed, what are those genes contribution to tolerance the plants against the biotic and abiotic stresses. These are the challenging tasks to identify. So before going to that, RT-PCR really into the how the gene expression is quantifying. So you have to know the basic principle of polymerase chain reaction. The earlier speaker also emphasized briefly about the polymerase chain reaction. So I will also just give you a glance about the polymerase chain reaction. It is an in vitro technique used for molecular biology to produce multiple copies of fragments of our DNA, especially for our targeted gene product. So earlier people were used 3 water baths at a time. So to maintaining the different temperature, to amplifying temperature, denaturing temperature, and extension temperature. For all those things, people were used the what we call the traditional instrument. This is called the water baths. So collective of this one is the right side one, which one you are seeing is the polymerase chain reaction machine. In this, all the thermal controller is there for the application of the genes. This technique was introduced by Carey Mullis in the year 1983. For this amazing contribution, Carey Mullis awarded a Nobel Prize in the chemistry in the year 1993. So what are all the PCR can do? Starting with 1 copy of DNA fragment, almost an infinite number copies of copies you can multiply with the machine thermocycler. The amplified fragments of DNA can be sequenced, cloned, probed, or sized using electrophoresis. Means after amplifying that part of your PCR product, how it is sequenced. So what are the sequences are present in your selected genes, and cloning for further characterization, probed, all those things can be possible with this PCR. Defective genes can be amplified to diagnose any number of illness. So let's say people are suffering from so many diseases, cancer and other things. So for those genes also you can quantify with this PCR. Genes from pathogens can be amplified to identify them, such as HIV, Vibrio, Salmonella, etc. So amplified fragments can act as a genetic fingerprints. So all those things can be done with the PCR. So coming to this PCR, what are all the components are present in this PCR? So let's say, for example, the component is 1 by 1. The component is highly required. One is the first and foremost is your DNA template. So second one is forward and reverse primers, and third one is the DNTPs. Then thermostable polymerase enzyme. This is most important because all the enzymes people we regularly it is used at the maximum at the 37 degree 35 degrees the maximum activity it will show. So for this PCR it will reach up to 96 to 98 degree to denature the what we call our DNA, double stranded DNA. After unwinding also your polymerase should be in working condition. So that's why it is a thermostable polymerase enzyme that is generally called as tag polymerase, bivalent magnesium ions and buffer. All these components are well required for the amplification of DNA fragments. So coming to this means DNA, you know that a DNA fragment you have to isolate your own source of the DNA, then forward and reverse primers, how to synthesize those primers. Primer means a countable number of nucleotides from 20 to 25 numbers. So all those you can synthesize using the bioinformatic tools. These are all the some of the tools available in the online for the free. Using these tools we can design the primers. In that again types of primers. What are all the types of primers? Gene specific primers, oligo DT primers and random primers. Gene specific primers are those which bind the specific target genes in a DNA. That is generally whatever the DNA you have to amplify that genes, you have to specifically synthesize the gene specific primers. Then oligo DT primers, these are all the short stretches of oligonucleotides. It made up of with T, that's why it is DT. T means thymine deoxyribonucleotide, which are used for cDNA synthesis from RNA as it binds to the poly A tail. So I will come later in detail what is this oligo DT. Then random primers. Random primers are those which binds to any region of DNA and non-specifically amplify different sizes of fragments from the template DNA. So what are the steps involved? Already Sir was emphasized in earlier talk. So there is a denaturing step, annealing step and amplification step. In this denaturation step, the DNA is unwind at the temperature 94 degree. Then annealing will take place depends on your what we call temperature of gene space amplification temperature of your new primers. Then extension at 72 degree temperature. This is the process. In this process, you can synthesize your gene product n number of copies. This you can see the exponential graph of the PCR product. Two becomes in one cycle it is 2. The second cycle it will go for 4, and again the third cycle 16 copies. It will multiply 2, 8, 16, 32, 64. It's like it will multiply. It generally for this people will suggest 30 to 35 cycles only. Why it is 30 to 35 cycles? Why not beyond that? Because what are the PCR components you put in the reaction mixture, those things will be stable up to 3 hours. Whatever you are using tag polymerase, it is a thermostable enzyme. It will be very active up to 3 hours. After that, it will it will reduces its activity. Then this is the final step. This is the PCR. After PCR, you have to identify that PCR product using agarose gel electrophoresis. After that agarose gel electrophoresis, you have to add the ethidium bromide. So ethidium bromide, it will bind to the double-stranded DNA. So it will, after exposing into the UV light, you will see the band. Means expression amplification of your what we call gene product. Then if you see this picture, this is the final product of your PCR product. This is the basic principle of PCR. So now we will see the applications of this PCR means. So molecular archaeology, molecular epidemiology, molecular ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery. Coming to the sequencing, bioinformatics, genomic cloning, human genome project, and also genetic engineering, site-directed mutagenesis, and gene expression studies. Now we are here, gene expression studies, how to study the gene expression, all those things we can see in the real-time PCR or RT-PCR. So now we will see the real topic of RT-PCR, means reverse transcriptase PCR. What is this reverse transcriptase PCR? So generally, so the reverse transcription polymerase chain reaction is called, otherwise called as RT-PCR, is one of the many variants of polymerase chain reaction. Earlier, sir explained that PCR made many things. Hello? Hello?
Sir, your audio is good, but your video is not moving. Your presentation is not moving.
Presentation is not moving. Okay, sir. Okay, sir. Now, which slide I am seeing that reverse transcription polymerase chain reaction? Same slide or other?
Hanging on first slide itself.
Okay. So I can visible, but why not? Sir, once again I will reconnect, sir. This slide sharing only. Yeah, 1 second. What up? Sir, shall I go with audio? So it's not connecting properly. I don't know what's the problem with this.
Sir, you continue, sir.
Oh, yes, sir. Yes, sir. I am connecting. So reverse transcriptase PCR. Sir, I will move with audio only. So I don't know why it is not
Okay.
sharing.
You
I
continue,
don't.
sir. You continue, sir.
Okay, sir. Okay, sir. So reverse transcriptation polymerase chain reaction is one of the many variants in polymerase chain reaction. This technique is commonly used in molecular biology to detect RNA expression. So how this will be means RNA PCR, RT-PCR is often confused with the real-time polymerase chain reaction, otherwise called as qPCR. How that confusion means? RT-PCR means using the reverse transcriptase enzyme for the PCR, that is called RT-PCR. RT-qPCR means the already synthesized using RT means reverse transcriptase, the whatever the synthesized cDNA, you are quantifying that cDNA using the PCR. That one it is called RT-qPCR. So RT-PCR is usually quantitatively detect for gene expression through creation of complementary DNA, that is cDNA transcripts from the RNA. So coming to this RT-PCR, there are two steps of RT-PCR types. One is two-step RT-PCR, another one is one-step RT-PCR. So in the two-step RT-PCR, traditionally RT-PCR involves two steps, the RT reaction and PCR amplification. So that is two steps. Means first you have to prepare the cDNA using reverse transcriptase enzyme, that is RT reaction, and the other using cDNA you are amplifying your gene of interest is called PCR amplification. So in this RNA is first reverse transcribed into complementary DNA using an enzyme reverse transcriptase. The resulting cDNA is used as templates for subsequent PCR amplification using primers specific for one or more genes. So this is two step. One is RT reaction, another one is PCR amplification. Now the one step reaction, the RT-PCR can also be carried out as one step RT-PCR in which all the reaction components are mixed in one tube prior to initiation of the reaction. So in this one step means the RT enzyme also you have to add in the reaction mixture, and also PCR components. I already told that PCR components are DNA template, primers, polymerase, tag polymerase enzyme, buffer, and DNTP. So all those things also you have to add, provided for the RT reaction. For RT reaction, what all you need? Oligo DT, reverse transcriptase, and other buffers. All those things you have to add in single tube, that is one step PCR. There you will have the master mix. In that master mix, according to the temperature, the enzymes will act and they will synthesize the product. In coming to our pandemic situation, COVID-19 measurement using RT-PCR, that is one step RT-PCR mix. So in this one step, all the required components they will add into the PCR tube, and in that what we call, they will take the swab. In that swab, they will isolate the RNA. That RNA they will add into that tube. That RNA will synthesize cDNA. That cDNA again it will amplify with the real-time PCR. So there it will give the product. That is one step PCR mix and the two-step PCR mix. Coming to the two-step PCR mix, so usually for two-step PCR mix, scientists will use, especially for the plant scientists or the animal scientists. First, they will synthesize the cDNA. Using that cDNA, they will do. They will use that cDNA further their work. Let's say, for example, that cDNA they will use for the construction of cDNA library. That cDNA they will use for the specifically amplify their gene of interest, and that cDNA they will use for further their interest in their work. So coming to the construction part of cDNA library construction, people will use that cDNA for the construction of cDNA library into 8 steps. So one is extraction of total RNA from the control and the treated leaves, and another one is purification of mRNA from the total RNA, then synthesis of control cDNA, and using the biotin labeled oligo DT. So means biotin labeled, using biotin labeled oligo DT, people will synthesize the cDNA of the control plant and also the treated plants. Subtractive hybridization between control standard cDNA and the stress mRNA, and selective precipitation of stress specific mRNA. Let's say, for example, you want to isolate stress specific genes. For that one, what you have to do, first you have to make the cDNA of the control and mRNA of your stress. mRNA of your stress are the treated one, and control cDNA, that is that cDNA and control means the stress mRNA you have to hybridize. So the common genes will hybridize. The only specifically represented, expressed in the stress condition, those will not be hybridized, because of in the control those genes are not expressed. Only stress specific genes will be not hybridized. It will be retained as such as mRNA of stress mRNA. That stress mRNA you have to isolate using biotin labeled magnetic beads. In that magnetic beads, you have to minus the what we call hybridized cDNA, hybridized stress mRNA and the control cDNA. Only stress specific mRNA will be retained. That stress specific mRNA again you have to clone into the suitable vector, and you have to create the cDNA library. That library you have to screen and you have to give for the sequencing. After sequencing those genes, means whatever the stress cDNA, if you cloned into the units of XR vector or whatever it may be, the size will be the bigger of your stress cDNA. That cDNA you have to clone it into the what we call a specific vector. In that vector you will get n number of colonies. Those colonies you have to pick, and again you have to isolate the plasmid. Thus plasmid, if you give for the sequencing, you will get the sequence. That sequence will be stress specific gene specific sequences. Those sequences, if you classify, you will get the what we call what these genes, where it is involving, where it will be functioning. If it is not, then it will give as a novel genes. So those will be, you can functionally characterize. After functionally characterize, then you have to go for the further characterization using real-time PCR. Now you have the cDNA of stress specific. That stress specific is irrespective whether it is a drought stress or salt stress or temperature stress or even chemically induced stress. So that cDNA library is, let's say for example, that is for water stress. So that water stress responsive genes, if you want to functionally characterize with other stresses, means your gene is specifically water stress gene. So that gene, whether it is expressing in other stresses, let's say for example salt stress or temperature stress or any kind of dehydration stress, that is called crosstalk. Crosstalk means the specifically water stress induced stress that is also expressing in the salt stress, that is also expressing in the temperature stress. That one you want to see, means again you have to induce the plants with different stresses. Those are all called as salt stress, water stress, temperature stress, dehydration stress. Then you have to isolate the cDNA of all the what we call stress induced plants. Means salt stress induced, you have to isolate the RNA and you have to prepare the cDNA. Then temperature induced plants, you have to isolate the RNA, then you have to prepare the cDNA. Then dehydration stress, you have to isolate the RNA and you have to prepare the cDNA. And irrespective, means like that you have to prepare the cDNA and use the gene of already you have isolated from the water stress. It is from your library yielded gene. That gene, if you use that gene specific primers, if you amplify using these many stresses, you will get the amplification, transcript amplification, how much expression is there in the water stress, how much expression is there in the salt stress, how much fold increases in the temperature stress, how much fold increases in the dehydrations. Like that you can functionally characterize whether these genes are specifically water stress genes, or these genes are specifically salt stress, or they are crosstalk between the both the stresses, means stress specific genes. Like that you can characterize. So coming to the application of this RT-PCR, this can be used in research methods. For example, qRT-PCR measure the expression of genes. Means I already told, so many crosstalk genes you can quantify using this real-time PCR. Then genetic disease diagnosis. RT-PCR can be used to diagnose the genetic diseases such as so many syndromes like cancer-inducing genes and syndrome-inducing genes, and also some other genes, means health-related genes. You can easily identify, easily quantify using this real-time PCR. And gene insertion. RT-PCR can also be very useful in the insertion of eukaryotic genes into the prokaryotics. Like that you can play with the what we call from prokaryotic system to the eukaryotic system using this real-time PCR. And also cancer detection. So scientists are also working on ways of RT-PCR in cancer detection to help to improve the prognosis and monitor the response to therapy. So many cancer-inducing genes you can identify with the real-time PCR. And RNA extracts. So RT-PCR is also used to test the RNA extract from different tissues, also for the presence of a particular transcript in order to determine the expression pattern of a gene in different organisms. So with this brief introduction, so I cannot share my slides. So I will stop my talk with here. So thank you very much, Ahmed sir, for the opportunity, and I am apologize. I am not able to share the screen properly, and I am not justice for the audience.
Sir, it is all right.
Hello.
I can understand your situation because you are in election duty today and you are there.
Yes, sir. Yes, sir.
And so many busy assignment schedules you had, and by overcoming all the hurdles, you are there in the meeting, and you have tried your best and
Level
effort
best.
to
Yes,
make
sir.
our meeting successful. So I express my heartfelt thanks to you. Uh, Now I would like to come to conclusion of today's conference. Aruna ma'am.
Yes, sir.
Shall we proceed to end the meeting?
Yes, yes, sure.
Yeah,
We have
I would
finished
like
all the
to.
talks.
Yeah, I would like to express my heartfelt gratitude towards today's speakers, Dr. Mehtab Bamji ma'am from NIN, National Institute of Nutrition, and Dr. Sham Kumar Bhandari from United States of America, Dr. Santosh Kumar from Thailand, and Dr. Insaf Ahmed Qureshi sir from University of Hyderabad, Biju Dharampalan sir from Kerala, and Dr. Harish Handral from Singapore, and Dr. Dipeswamy sir with all his efforts. With all these efforts, all the speakers made today's meeting very successful, and on behalf of the Telangana University and Department of Botany, I would like to express my sincere thanks to all the people who gathered over here, participants also. I request once again participants, while filling the feedback or by filling the form, be careful about the emails, because morning we were communicating our today's meeting link, and some people like Rakshita and some BS Anusha, Deepika, Vasuki, these people had, and one more person I couldn't spell out the name, were having their names, emails given misspelled. So please, while filling this, you be careful and fill it properly. So with this, we come to an end of day one international conference on emerging trends in life sciences. And here I express my deep sense of thanks towards the registrar, Telangana University, Professor Naseem ma'am, and my esteemed fellow colleagues from the department. Also, I express my deep sense of gratitude towards them for making me, encouraging me to making this event successful. I express my heartfelt thanks for Professor B. Vidyardhini Madam and Dr. Professor M. Aruna Madam and Dr. Srinivas and Dr. Jalandhar, and our public relation officer, Dr. Triveni Madam, and Mr. Naveen. He's computer and computer programmer who looks after the website. So all these people who are the cast and crew of this entire conference, I express my thanks, and expect that tomorrow also all the participants will join in the same platform, and the link will be shared in WhatsApp group, and will be sent to your email IDs respectively, what you have submitted in your registration form. With this, we declare day one to come and end. Thank you very much. Thank you one and all for joining us. Tomorrow again you join us. If you have any queries, please you can drop your queries to our email ID given, and emails are sent to you timely. And if you have queries, we'll forward the queries and your email ID to the respective resource person, and you can get your query answered. Thank you very much one and all. And